But you know what's interesting with the Ifakara is that somehow history has conspired such that we have been part of nearly all the malaria vaccine candidate tests.
That's not history conspiring. That's investigators using the best available clinical team at site. So sure.
Yeah. So I mean, we were part of the Passaroyo vaccine trials.
Yeah, I remember.
And we've gone through that up to now, the new generation vaccines. Adrian, if you don't mind, I'm just going to start admitting persons. Is that okay?
Of course.
All right. There is approximately 80 people on the line right now.
Good.
And we have about 300 registered, so we expect about half of them will join live.
Oh, good.
And I'm just starting now to admit them. So you guys getting busy again? You were busy during COVID and starting now monkeypox?
Yeah, busy as normal. Yeah. So no, monkeypox is not impacting on us. It's you know, there is a licensed vaccine. It's being distributed. It's just scaling up the manufacture, and that's not being done here this time. So yeah.
And that is just the original smallpox vaccine, or is it?
Yeah, it's a particular type of smallpox vaccine that has been attenuated by over 500 passages in the lab. It was made in Germany in the 1970s and 1980s, and is mainly used as a recombinant vector for malaria, HIV, TB, and so on in clinical trials. It's not licensed for any of those, but the actual non-recombinant viral vector works against monkeypox and works against smallpox. And that is licensed by a Danish company.
Yeah, we have at least one question around that actually.
Sure.
Hopefully we can discuss that a little bit. I just want to inform you also, Adrian, that the masterclass is live on YouTube now.
Good.
So we try to do that just in case there are people who are unable to join us. Sometimes people register and then something comes up. So we try and keep that. Also, this is the 38th edition of this event.
Excellent.
And we started in late 2020, I believe, in the beginning of 2021. And I think we've had a good run so far. We've been lucky to talk to quite some great experts around the world. And we ask a lot of questions, so hopefully you don't mind.
I think that's the plan. Yes.
Yeah. So, ladies and gentlemen, thank you so much for joining with us today. It's fantastic to see you all for the 38th edition of the Ifakara Masterclasses. We're joined today by the great Professor Adrian Hill from Jena Institute. I don't think you get anyone better to talk about this subject than Adrian, and I think we are very, very, very lucky that we have this chance. We were going to have this conversation with him much earlier, but because of commitments, we've been shifted multiple times, and finally we are able to have the conversation today. We will be joined shortly by my colleague Ali Olotu. Ali Olotu from Ifakara is joining the conversation today as well, both as a co-host and also to contribute certain aspects of the conversation. My regular co-hosts, Dr. Sheila Ugoma and Dr. Nana Abba Williams, are on duty travel. They will be joining us in the middle of the conversation, but even before that, we can begin. So I hope that you guys are ready to go, and please, as usual, ask as many questions as you wish to ask in the chat. If there are any subjects that you want to contribute to, please let me know, and you can do that by either asking myself directly, direct chat message, or to my colleague Halfan Gowo. Halfan Gowo is on the line here, and is going to is supporting also as a co-host, trying to if, for example, there are any specific concerns that you might have, Alfan, I'm going to request you or any of my colleagues from IFCARA to post the link for the YouTube channel here, just in case someone wants to share this with their friends. And also, please feel free to share the link to the masterclass to your other colleagues who might not have received the link and are willing to join in today. So I think, Adrian, what might be really, really nice so first of all, ladies and gentlemen, just be aware the conversation is live already on YouTube. And we will have a slightly different style today because of the technical nature of this subject. We've requested Professor Hill to begin by giving us an overview of the work on malaria vaccines and also a little bit on SARS-CoV. So we will begin briefly with that for the first 20 minutes, and then we continue with our the usual style of our conversation. So I think, Adrian, I'm going to invite you right now to just begin that as we welcome the rest of the participants. Thank you so much and welcome.
Excellent. Thank you, Fredros. And thank you very much for inviting me. My only regret is I didn't give this talk 2 months ago when I was in Tanzania, in Bagamoyo. So it's a pleasure to speak to so many people online. And thanks for covering malaria vaccines as a topic. It really is at a very exciting stage. Those of us who've been involved in malaria vaccines since really the early 1990s. There's never been a time of more promise and more excitement about what's happening with malaria vaccines and what they can do to help control malaria and eventually eliminate and eradicate it. So I'm going to speak briefly about malaria vaccines as we discussed, but I'm going to start with one slide on COVID vaccines, partly to remind you that a lot of what we've been doing in the last few years in Africa in a phase 2b trial and a phase 3 trial has been against the backdrop of COVID and a global public health emergency and the biggest pandemic in a century. But somehow we've managed to make progress nonetheless on malaria vaccines, as I'll explain. But let me start with a slide that comes from a journal called The Economist that some of you might see, enumerating the number of lives saved by COVID vaccines in 2021. So that doesn't count what vaccines are doing this year in 2022. And the numbers are kind of interesting and surprising. So we were pleased in Oxford because we were involved in designing this vaccine and developing it, that the AstraZeneca Oxford vaccine, by the best estimates of people like Azra Ghani at Imperial College in London and others, had saved just over 6 million lives through vaccination against COVID-19, closely followed by Pfizer-BioNTech, around about 6 million, and then nothing else with half that number. Sinovac, maybe 2 million, Moderna, less than 2 million, Sinopharm, maybe 1 million. Then the Russian vaccine, Sputnik 5, Johnson & Johnson, Bharat Biotech from India, CanSino, and other vaccines. And the big red arrows on the side are to show that 4 of these, more than any other technology, were recombinant viral vector vaccines. In fact, all adenoviral vectors, whether made in China by CanSino, in Russia by Sputnik, in the Netherlands by Johnson & Johnson, or at 20 sites around the world by the AstraZeneca Oxford Serum Institute team. And that is partly to make the point that you can make and distribute vaccines at very low cost. That's what we were doing with the Oxford AstraZeneca vaccine, and doing it fairly quickly, as you can see there. That was the year following the discovery of the virus. But also to highlight that very different yields come from different manufacturing processes. And I know a lot of you are interested in vaccine manufacturing in Africa, which is an extraordinarily important issue. But just note here that the biggest pharma company in the world, Johnson & Johnson, which does adenoviral vectors very well and has done them for years and has a licensed Ebola vaccine, were able to make what barely 1 million vaccines to save barely 1 million lives. The Russians, who hadn't done so much previously, slightly more perhaps with Sputnik 5, but an academic group led by Sandy Douglas here in the Jenner Institute in Oxford came up with a process that was not only transferable to 20 facilities around the world, but was remarkably high yield, thanks to a simple improvement now published, discovered in the early months of 2020. And that changed, I guess, our impact from maybe 1 million to 6 million lives. So manufacturing really matters. Unfortunately, it doesn't get into nature. It should. It's a very important activity, but keep in mind that you can only get impact if you can manufacture lots of vaccines. And this is super relevant to monkeypox, which you mentioned a few minutes ago, Fred Ross. We can come back to that. But the other reason for showing this slide is there are actually 3 reasons why doing this with the AstraZeneca Oxford vaccine was only possible because of malaria. And what those 3 reasons are will be mentioned as I go through the malaria slides in the next few minutes. Look out for them, because I'm going to ask you what they are. The clues are all in the talk. And malaria played a really important part in us being able to make that COVID vaccine. So you know more about malaria, most of you, than I do. Not a good position in 2020. We don't have the 2021 data from WHO yet, but deaths increased undoubtedly in 2020 to an estimated 640,000. That number was below 500,000 a couple of years earlier. And of course, these are mainly deaths in childhood, mainly in Africa. And on top of that, you've got hundreds of millions of clinical episodes, which are no fun. If you've had one of those, you'll know. We have drugs and insecticides and other interventions, obviously bed nets, that are becoming a bit less effective by and large. We've had those for decades. We are spending over $3 billion as a global community on trying to control malaria with these tools. I would argue that we desperately need new tools, and a vaccine is probably going to be the most effective in say, the next few years. But some leading vaccine candidates have been struggling. There's far too little money for malaria vaccine development. I calculated last year that 2 days spent on COVID vaccine development was about the same amount of money as every year spent on malaria vaccine development. So the field is hugely underfunded. RTSS from GSK has been the leading malaria vaccine for about 20 years. It isn't licensed yet. Hopefully it will be very soon. But that was delayed from 2015 to next year in its deployment by a blip, really, in the phase 3 data. And we'll come to that in a moment, which was very frustrating. And of course, you know, in terms of resource allocation, there's no doubt that there were more malaria than COVID-19 deaths in Africa in 2020, yet the resource going to COVID-19 in Africa alone was far greater than for malaria. This is just my way of saying that malaria vaccine development has not been easy, as COVID vaccine development was. COVID took months. Malaria started back in 1910. And is still going, waiting for a licensed vaccine. And of course, a parasite is much more difficult to vaccinate against. There are no human parasitic, antiparasitic vaccines. But this is an example of what we're doing here locally in Oxford. We have clinical development programs on all 4 stages of the life cycle. I think we're going to need most of those vaccine types to eradicate malaria. I'll talk only about R21 today because that's the most advanced of these candidates, and just in passing mention that for many years we worked on viral vectors, including doing the first chimpanzee adenoviral vector trial back in 2007 for any disease here in Oxford. We do get efficacy with viral vectors against the liver stage, but it's not as high as is needed. It's maybe 25% sterile protection, 50% or so partial protection. Blood stage vaccines are making a late run, doing really well, particularly with the Rh5 antigen, now in clinical trials, now showing efficacy in human challenge and in trials in Africa. And one of our plans is to put Rh5, that antigen was discovered by my colleague Sandy Douglas, who I mentioned already, working in Simon Draper's group. Simon is leading this blood stage program as a whole, and combining Rh5 with R21 may be a particularly effective combination for the future. And then transmission blocking vaccines are getting rather exciting, because just in the last year, in fact, in October of last year, there were the first data from Mali, working with Patrick Duffy's group at the NIH, on efficacy against transmission in humans. That's with the PFS 230 antigen. The PFS 25 is looking quite exciting in the hands of my colleague Shuni Bijwas here, who's leading the Oxford transmission blocking program. She has 25, 230, and 48, 45 in clinical trials. But it looks to me as if transmission blocking vaccines might be relatively easy, because believe me, all the other stages are not, and people have been working on them for decades. So let's start with the leading candidate for decades, as I've said. If you can read the publication date here back in 1988, that's how long RTSS has been around. And the idea is very simple. The circumsporozoite protein that coats the sporozoite of Plasmodium falciparum has a central repeat of 4 amino acids repeated 20 to 30 times, and antibodies against that central repeat protect against infection by binding to the sporozoites soon after they're inoculated in the blood and stopping parasites getting into the liver. If no parasite goes into the liver, there'll be no malaria. So in principle, that's very easy. In practice, it turned out to be very difficult, because the first generation and even the second generation anti-CSP vaccines did not show efficacy. And it wasn't until Joe Cohen in Belgium back in the late 80s had this neat idea of fusing that antigen to the hepatitis B vaccine that SmithKline Beecham were selling at the time, then a new vaccine. And that, of course, made the antigen into a nanoparticle with malaria on its surface. And nanoparticles are, of course, far more immunogenic than soluble proteins. You get higher antibody levels, and that was the key. If you get enough antibodies against the NAMP, you will block all of the sporozoites getting into the liver and prevent infection. Sounds simple. In practice, it didn't work out terribly well. RTSS went through phase 1, phase 2, and then a large phase 3 trial in Africa in nearly 16,000 children of different age groups, young infants and older infants and toddlers. With 3 doses of vaccine, the highest efficacy was 28% over 4 years of follow-up in the phase 3 trial. But with a 4th dose, you got up to 36% efficacy over that several years. And the health economists were all agreed that it was clear that that was cost effective, not because it's the best vaccine in the world, but because the impact of malaria is so great that cutting down disease and deaths by around about a third would be cost effective. So that went forward for licensure by GSK, firstly to the European Medicines Agency, which were positive about it and gave a positive scientific opinion in July 2015. But then when WHO came to review it for pre-qualification, the SAGE committee of experts were concerned particularly about a safety signal. There were, for some reason, more female deaths in the vaccinees with RTSS than the controls. And that was not a trivial p-value. We now know that that almost certainly occurred by chance. The problem was it took implementation trials to the scale of 800,000 children organized by the WHO over the next 5 years, with the data really only coming out last year, to show that in 800,000 subjects, vaccinees and controls, there is actually no increase in female mortality. And hopefully that vaccine it's already been approved for a policy recommendation by WHO, and hopefully the pre-qualification is imminent and there will be vaccine doses next year. So what we did in the meantime, and this was work of a graduate student in the lab here originally, made R21, which is similar to RTSS, except that RTSS has 1 molecule of RTS, the repeat, the T cell region, and the surface antigen, for 4 molecules of the surface antigen. So only 1/5 of the molecules in the nanoparticle are actually malaria. That was because in the 1980s, there weren't promoters, there weren't yeast strains that allowed you to express only the fusion protein. By 2012, we succeeded in doing that with a different yeast strain, and we now have a nanoparticle that is coated with malaria on the surface. So in the cartoon form, this R21 has CSP on the surface, particularly the NAMP repeats, whereas RTSS is mainly covered with hepatitis B surface antigen. If you take a monoclonal antibody to hep B, you can stain this particle very well. You do not stain R21. And there's a very brief summary of some differences here. This is obviously more recent, a different strain of yeast, which was important for the manufacturer. I've talked already about the ratio of S to RTSS, and spoiling the story slightly, we see high-level efficacy in African children repeatedly with this immunogen, and we're now targeting licensure next year. The other important difference is there's a simpler adjuvant here, Matrix M from Novavax. It's licensed for COVID now, that we're using with R21, whereas GSK used their AS01 adjuvant that also has a monophospholipid A component as an adjuvant in addition to the saponin. So we were very pleased when we took that into the clinic for the first time here in Oxford and did our usual mosquito bite challenge study with the controls, either all coming down with malaria or maybe one not within 2 weeks. And the vaccinees, particularly in a low dose group, 3 doses of just 5 micrograms or 5 to 10 micrograms of R21, 9 out of 11 were sterilely protected in the first clinical trial. That encouraged us to move forward quickly with our new industrial partner, the Serum Institute of India. I'll come back to them later. They were very keen to take this forward and manufacture at scale. And the next trial was actually in Kilifi in Kenya. It was a phase 1B age de-escalation trial that showed good safety and high immunogenicity in Kenyan adults, children, and infants. That led on to this phase 2B trial led by Haladu Tinto, in Nanoro in Burkina Faso at the CRUN unit. A very simple design, 3 groups of children aged 5 to 17 months of age. The control group got a rabies vaccine, no malaria vaccine. The vaccinees either got 25 or 50 micrograms of the adjuvant, but they all got the same low dose of the antigen, being R21. So we're really testing here if more adjuvant would make a better immune response. 3 doses, 0, 4, and 8 weeks after the first dose. And this is in northwestern Burkina Faso, an area of very seasonal malaria transmission. About 3/4 of the cases are from July to December, mainly in the middle of that period. And the other quarter of the cases are in the rest of the year. So I've shown you that. The immune responses were interesting. With the higher dose of the adjuvant in group 2, you get about double the antibody response. And this is crucial because higher antibodies are more likely to protect you from malaria. And believe me, these are very high levels, hundreds of micrograms per mil of antibody. They do drop, however, over the subsequent year. And that's why you need a booster dose at a year, a 4th dose, if you like, and then the antibodies come back to similar levels to what you got after the primary series. And these, interestingly, these antibodies are about 6 times higher than you see in UK vaccinees, who, remember, were largely protected against malaria. So what about protection in the field? Well, the good news here was that with, with a year's follow-up, these are the controls, most of them getting malaria. These are the vaccinees, slightly fewer in the group 2 with the higher adjuvant dose, and 77% efficacy overall in that group over a year. So that was exciting because that was the first time that any vaccine in a field trial had met the WHO's target of 75% efficacy or higher in African children. And though we got those first data from this trial showing efficacy in January of 2020. How does that compare to RTSS? Well, at the same site, RTSS was tested in their phase 3 trial. The point estimate was 44% efficacy in contrast to 77% over a year. But importantly, RTSS was given at any time of the year in the RTSS phase 3 trial, and that may account for, I guess, about half of the difference in efficacy here. So it's partly with seasonal vaccination being better, and it is for RTSS, and it's partly, we believe, with a better vaccine. So what happened after that? Because more than 1 year's protection is required. So after the 4th dose, if you simply count the episodes in group 2 and in group 3 over the 2nd year, you can see they're different. These were similar sample sizes. So again, there's very good efficacy, 77.5% efficacy in year 2. That's not published. It's coming out in the Lancet Infectious Diseases in the next month. And if you put the 2 years together, the efficacy is, as you would expect, 77% in reducing episodes of malaria. That encouraged us and the Serum Institute of India, who have funded the phase 3 trial, to go forward into this large phase 3 trial in 4 countries, in Africa, in Bagamoyo, in Tanzania, in Kilifi, in Kenya, and then most of the subjects in West Africa, in Nanoro, again, in Burkina Faso, but again, down in Dande, where there's more non-seasonal transmission or perennial transmission in Dande and Burkina Faso, and importantly in Buguni in Mali. So this trial started at the beginning of 2021 in Q1 with the first dosing. It's been running since then, initially at the seasonal sites and then at the standard regimen sites, as we call them. A big difference here is that we vaccinated not 5 to 17-month-olds, but 5 to 36-month-olds, because of course 1 and 2-year-olds die of malaria, and you might want to vaccinate them if they miss out on infant vaccination. The trial is sponsored by the clinical sponsor, University of Oxford, funded by the Serum Institute of India. And the primary endpoint, coming up fairly soon, is efficacy at 12 months of follow-up. So where we are, and I'm about to finish on R21, is that we have the 2-year data I've just shown you on phase 2b. We'll have the 3-year efficacy data very soon in the next month or so. The phase 3 trial, of course, is fully enrolled. We'll have the primary endpoint later this year. And Serum Institute have shown that they can easily manufacture this vaccine efficiently and can produce hundreds of millions of doses a year if that is required. In terms of regulatory approval, we were very pleased that the WHO unusually granted us permission to apply for both a policy recommendation and pre-qualification in parallel from the end of September this year, presenting the data that we have at the time. By the end of September, we will probably continue to provide data over the coming months and years after that, and it's up to the regulators to decide when they have seen enough to convince them to go forward to pre-qualification. But we have been making the point that there are far more malaria deaths than COVID deaths in Africa now and have been for the last few years. So why should a malaria vaccine, which was licensed in weeks to months around the world, receive such, COVID vaccine was licensed in weeks to months around the world. Why should a malaria vaccine in Africa not receive the same priority review? So just to summarize really the advantages of R21 that we see, we have higher efficacy in our phase 2 trial. Phase 3 data are imminent. We're using a lower dose of the vaccine, 1/5 of the dose, which makes it less expensive. Of course, there are other reasons it's less expensive. There's a less complex, less expensive adjuvant being used. That is Matrix M, that's now licensed for COVID-19. A better safety profile, at least in our phase 2 data. But none of those are really the most important point. The most important point is that with R21, it will be possible to supply 100 million, maybe 200 million doses next year. Whereas for GSK, they are struggling to produce 7 to 10 million doses. Now, as you know, there are about 40 million children born each year in malarious areas of Africa who could use a vaccine. If they need 4 doses each, that's 160 million doses. So if you only have 7 million doses, say, for RTSS, you've got a huge distribution challenge. So hopefully R21 can help with that. So I've talked about the scale of supply just now, given you that calculation. We're hoping to do catch-up campaigns as well as primary series vaccination in infants. Funding is needed either from Gavi or the Global Fund or from somewhere else, because 200 million doses at $3 per dose is a lot of money. But remember, there are $3.5 billion being spent on malaria control already. And we do need timely review and approvals. We can't wait 5 years while that child is dying every minute of malaria for getting the requisite approvals. So I hope that we don't have to. So big picture, we think that new vaccines, not just R21, but other ones coming along this decade, should be able to reduce annual deaths by about 90% by the end of this decade. I don't think that's a fanciful aspiration. You can calculate that that should happen with good vaccines. And then in the 2030s, we can get on with malaria elimination and eradication, hopefully finishing by around 2040. And I'll just stop by mentioning that dozens, if not hundreds of people have been involved in the R21 program over the last 5 to 10 years. These are some of the key people and the key phase 3 trial principal investigators, as well as Tinto in Burkina. We have John Bosco Ouedraogo, Alassane Dicko in Mali, Manga Hamaluba in Kenya, and of course, Ali Alutu locally. Thank you.
Excellent. Thank you so much, Adrian. That was a very optimistic note to finish upon. It's very rare that you have people who still believe that we can do something by the year 2030. You know, in recent years, we've been at this stage where it's mostly, the story is mostly about the failures in malaria control, or the weaknesses of really how much the fight is weakening. And here you are providing a very optimistic view of this, and thank you so much for that. You had a slide where you acknowledge all your colleagues, and I would just like to let, if any of your co-investigators is on the line and they would like to participate more actively on this call, please feel free, especially for the PIs, Alidu Tinto, Ali Olotu, and the rest. You may have the privilege to turn on your video because some of the questions coming up, perhaps it would be a good idea that you co-answer them with Adrian. So Adrian, I hope this is perfectly fine if we invite some of your co-investigators.
It would be an improvement, Fred Ross. Yes.
The other thing is there is now 200 people on the line, and we believe that there's many more people who are going to be watching this by you on YouTube. And I would like to invite anybody who has any questions, kindly put that on the chat, and we take it on from there. So, Adrian, we're going to move forward with this now. There's a lot of questions coming up as a result of your conversation, of your presentation, and there will be on the chat. So
okay,
and we can bring them together. You don't have to ask them now because a lot of them will be aligned to the questions that we want to ask you. But if there's anything specific that you see there that you'd like to address, you can address it at any time, because this is really your show. You're going to be speaking like 90% of the time, so you can merge any of the questions with whatever we ask you, and you can do that. So I'm going to share some of the slides here as well and see. Can you see my slides, Adrian?
Yes.
Excellent. So I think your presentation was an excellent start to the topic of today, which is the next malaria vaccine. And we had a great excitement in the year 2021 when WHO provided the approval for the RTSS. You've explained how different this is and how similar it is to RTSS. But we would like to start our conversation really with some of the work that you do, and not just on malaria, but on the other things as well. We will be covering a wide range of topics. We would like to look at how malaria vaccines will change how vaccines will change malaria control overall. We would like to look at some of the lessons learned from the RTSS, and we've covered a number of these already. What should be the next vaccine and what should they look like. And I think the R31 candidate offers us some great examples of that. So we will cover some highlights from that as well, in addition to what you have already described. And then at the end, you describe the partnerships that you have that enables this testing with the industry, with your colleagues in endemic countries, the great scientists who've been supporting this test and leading this test, and also with your own colleagues at Oxford. So we would like to talk about, how important these partnerships are in the fight. But Adrian, first of all, I mean, you are at Jena. Jena has a fantastic history with smallpox. Now monkeypox has just been declared a pandemic, one more time, public health concern. And it's probably a good idea for you to just briefly describe the work that you do on vaccines broadly at Jena. Of course, we know that malaria vaccines are the core of it, but we're also aware that you do a lot of work on SARS-CoV and other vaccine candidates. And if you like, you can talk a little bit about monkeypox as well. Go ahead, Adrian.
Yeah. So look, we're a university-based vaccine institute. We've been going since 2005. The reason we founded the institute then was that there were already 3 clinical programs on important disease areas. Malaria was the first in the clinic in 1999, followed soon after by HIV, led by Andrew McMichael and Tom Hanke, and then tuberculosis vaccines were coming along, and Helen McShane took her first subunit vaccine into an efficacy trial in South Africa some 5 years later. The reason for coming together was, of course, we felt that we could do more together by sharing technologies, by sharing understanding, experience of clinical trials, and so on. And that turned out to be the case, and the institute has grown really ever since over the next 15 years to embrace not just those 3 very important diseases, but other important targets like a better flu vaccine. Sarah Gilbert worked on that for quite a while. New vaccines, we have a program for gonococcal vaccination led by Cal McLennan at the moment. We have more difficult targets like cancer vaccination. Even vaccines against Alzheimer's and Parkinson's disease are being worked on by one small group. So we're not limited to, and of course, lots of bacterial vaccines, Andrew Pollard's work and so on. We're not limited to any particular disease. And if people can raise the funding and support a program, we're happy to support them. So what does the Institute provide that you couldn't get from your own research group? The answer is core facilities, and there are more and more of those. So one you probably are aware of is the viral vector core facility that made the COVID vaccine very quickly in January, February of 2020. That was about their 500th viral vector they had made, because all the others had been made for other programs, whether malaria or TB or flu or whatever. So they had a lot of experience doing that. And they make them very well because 5 people all day, every day make those vectors to a very high quality. And that would be a much better solution than a new graduate student coming and spending a year making their first viral vector. So that's one core facility. Another really big one is our GMP manufacturing facility, which we have here on site next to us. And that has made vaccines for the Jenner since 2007. The first one, incidentally, was a chimp adenovirus vector vaccine for malaria. A new core facility is an mRNA facility. Now, there's so much interest in mRNA vaccines. Lots of people want to make those, but the cost is quite high and the barriers to entry is significant. So we're helping with that and exploring new technologies like thermostable RNA vaccines that we think will be needed for many, many countries. And then that gets your program up to off to a good start if you can compare different vaccine types for whatever disease you want to work on. And you have the animal facilities, which we have next door, to test those in mice. And then you have preclinical data to support an application for clinical development. But overall, we've taken, I think it's 57 candidate vaccines from the bench into clinical trials in the last 20 years.
Do you guys work on, you guys have worked on Ebola vaccines as well, right?
To a degree, yes. We were not, in 2014, when there was a large West African outbreak, we had not been working on Ebola vaccines. What happened was WHO knew that we worked a lot on chimpanzee adenoviral vectored vaccines. There was a vaccine from NIH and Kairos. Kairos is or was an Italian biotech. They had made a chimp adenovectored vaccine for Ebola. They'd got it to GMP manufacture, but we were asked to do the clinical trial and get it moved to Africa as quickly as possible. And we did that in 2 months, in August, September 2020. And then we went and made an MVA vector to boost that immune response and make sure we'd get even better immunogenicity. So we did make an Ebola vaccine then. Teslam here is now working on other Ebola vaccines. Some of you may know that there's a Sudan strain of Ebola. I don't think anyone has a vaccine in the clinic for that. If there's a Sudan Ebola outbreak next week, there's a problem with no vaccine. So we're trying to fill that gap. So yes, so what we did after 2014, when money became available for outbreak pathogens in large amounts, was we set up a group focused on outbreak pathogens, led by Sarah Gilbert, notably working on MERS, a coronavirus, and several other candidates. I think there are 7 or 8 outbreak pathogen vaccines we've made now, including chikungunya and Zika, and of course, importantly, working with George Warimwe from Kilifi, who was here at the time and is now running his own Rift Valley fever vaccine trial in Uganda. So that's just a flavor of some of the things that we do.
No, excellent, Adrian, and I think on behalf of all my colleagues on the call here and those who are not on the call, we just would like to say thank you to yourself and also to the Jenner Institute for the incredible platform that you're offering, for the support, making sure that these innovation processes are actually proceeding and that people are able to do what needs to be done to bring the diseases under control. Adrian, we would like to bring you back to your early days working in Africa, your early work in West Africa with Professor Brian Greenwood, who has also given us an early masterclass here. Some very interesting observations there. You published 2 papers in Nature, one in 1991 and a follow-up paper in 1992, where you characterize these HLA genes. Talk to us a little bit about how this story ended. First of all, how did it start and how did it end? I mean, this was a very interesting story back then.
Yeah, so I suppose it started when I did my PhD. That was in the mid-1980s in David Weatherall's department here in Oxford. He was a great human geneticist as well as professor of medicine, very interested in resistance to malaria because he worked mainly on globin genes. And of course, sickle hemoglobin and alpha thalassemia protect you against malaria. And my PhD was on alpha thalassemia in the Southwest Pacific, in Melanesia, mainly in Vanuatu. And we were able to show correlations between the prevalence of malaria resistance genes there and the historical endemicity of malaria. So for example, in Polynesia, where there's never been any malaria, there's no malaria resistance genes that are new. The link then really to Brian Greenwood was when I was looking for something to do as a fellow after that, after I'd done my sort of general medical training. I approached Brian and said, can we look at HLA and malaria in the Gambia in West Africa? And of course, the MRC unit had already been there for about 50 years. And there was a severe malaria study in progress, looking at risk factors for severe malaria, a very comprehensive epidemiological clinical study looking at many potential influencers. And the one I asked to add into that was human genetics and HLA. So we typed literally thousands of HLA complexes from children with and without severe malaria in Africa. And we found some evidence that HLA types, particularly HLA-B53, were associated with resistance to malaria in that population. And then that led on to work with Andrew McMichael, who was the doyen of cellular immunology, particularly CD8 T cells in the UK, and looked to see if we could find some CD8 T cells that were restricted by HLA-B53, and indeed we did, to a liver stage antigen. And that looked to us as if it could be the mechanism of protection and afforded by that HLA type. So the real importance of that is it started a lot of interest in liver stage immunity and attempts to make liver stage vaccines that would induce CD8 T cells. And that was a long road that started in sort of 1995 and ran for over 20 years, trying to make vaccines that would induce CD8 T cells with DNA vaccines that were then new. That was our first vaccine trial. With viral vectors like MVA, that was our second vaccine candidate for malaria. And the frustrating result, even though it was encouraging most of the time, was we could protect people against malaria, but not enough. So about 25% efficacy is not really a vaccine. We asked if they would work better in East Africa. We did one trial with the Khalifa unit, and there seemed to be higher efficacy with viral vectors, possibly because people in Africa have pre-existing T cells to liver stage parasites, and that's still running that program, but it was overtaken by R21 for the reasons we've just talked about.
Excellent, and you've maintained this partnership with your West African colleagues, and of course, as you've described, the R21 is a perfect example of not giving up on that.
And just absolutely, and just to add to that, the first efficacy trial of putting a blood stage vaccine, RH5 from Simon Draper's group, together with R21, is due to take place in West Africa, including the Gambia, for a safety trial in the next 6 months. So that's quite exciting.
Cool. Yeah. Excellent. And then broadly speaking, and my colleague Ali Oloto has now joined. Ali, we're going to invite you to turn on your video as well. Adrian, broadly speaking, and you touched a little bit on this, but please expand on it if you like. It's starting to look like the vaccines are actually going to be possible. You know, for a long time before the RTSS, it was very bleak. You know, at some point, I remember reading Malaria Corpus when I was a PhD student. You know, you kind of give up on this whole thing, but you guys kept maintained this effort, and now it starts to look like malaria vaccines can actually be a reality. Now, how do you think they will actually change the game?
I think it depends on how they are used, and this is where I'd really value feedback from this audience, because I didn't actually talk about how this vaccine would be used. So let me give you a few scenarios and see what people think. When RTSS was up for use in 2015, the general idea was you would vaccinate children at 5, 6, and 7 months of age and give them a booster 15 to 18 months later. And that was really what was tested in the phase 3 trial. And you saw the efficacy data. It's useful, but it's not high level. It was about 36%. So what else can you do? Well, I think the biggest innovation in the last few years has been seasonal vaccination. And that was really started off by work from Mali and the London School working together, and then with Burkina Faso, and then that excellent paper in the New England Journal of Medicine that came out, I think, last year, showing that you could get the overall efficacy of RTSS up to 62% over 3 years by giving the vaccine seasonally. And we understand why that is. If the antibody titers are higher when you get malaria, your vaccine's more likely to work. If you wait a year and they've gone down 75%, for example, then you've got lower antibody titers, you're more likely to get clinical malaria. Yeah. So there's been a huge amount of discussion about whether, at least in areas where malaria is seasonal, we shouldn't use seasonal malaria vaccination. And how would that be given on a campaign basis? Would it be a booster every June? Would it be EPI vaccines according to age, and then an annual booster for 2 or 3 years in just before the malaria season? That's one option. That's now, I'm told, being called the hybrid model. You'd get the EPI team to vaccinate every child at 5, 6, and 7 months of age, and then the booster would be before the rains or just as the rains start in West Africa. And that, of course, I think would make a great deal of sense. It would be more impactful. The question is how deployable it is and who would do it. And by the way, it is our favorite approach. So would it be the EPI program, Would it be the National Malaria Control Program who deliver seasonal malaria chemo prevention every year in so many countries? Would the same individuals be able to give the drugs later and give the vaccines in just before the rains? That all needs to be planned, and there are lots of discussions going on, I know, at WHO about doing that. So that's the second big variable, if you like, the scheduling of immunizations. But remember, what you're trying to do is very simple. It's keep the antibody levels high, and you can measure that in a trial of 30 people with different schedules.
Yeah.
And the third variable I'd value your feedback on is age of immunization and catch-up campaigns. Nobody talked about a catch-up campaign with RTSS, to my knowledge, and you now know why. There isn't enough vaccine to vaccinate half of the 5-month-olds, let alone start vaccinating the 1-year-olds and 2-year-olds. Now, we were aware that Serum Institute, with their manufacturing technology and newer yeast strains and so on, could produce hundreds of millions of doses. And by the way, they want to produce hundreds of millions of doses because the larger the scale of manufacture, the lower the price of a single dose. And the price has to be very low for a malaria vaccine. So that's a win-win for everybody. So would you just vaccinate the 5-month-olds and boost them later? Or would you vaccinate, say, everyone from 5 to 17 months of age? That was done in the RTSS trials and in our phase 2 trials. Or would you actually vaccinate the 5-month-olds and do a catch-up campaign to 3 years? We've shown in our phase 3, we think we've shown, data are still blinded, that you can vaccinate safely up to 3 years of age. So, you know, if children are still dying at 1 and 2 years of age, and the mother is watching the younger child get vaccinated at 6 months of age, if I were one of those mothers, I'd quite like my 1 and 2-year-old vaccinated as well. And if there's money to do that, why wouldn't we do it? It's probably cost effective. We can do those calculations. So, you know, the other question then is, do you do a big catch-up campaign once you have a vaccine that's, you know, over 70% effective because it's cost effective?
I mean, interesting points. And as you see on the slide here, we have some suggestions that align very well to the question you're asking back to us. When I first asked you this, the first thing you asked was, it depends on how it's used. You have here Professor Melissa Penny and the team at Stockholm Public Health Institute describing some potential use cases of vaccines, and they highlight here the question of all-age vaccination, the combination with the SMC. It's great to hear that, and also as you describe what is going to happen with catch-up campaigns should those be rolled out. I would like to ask Ali. Ali, would you like to add anything on this, or ask? Well, I mean, Adrian asked us back this question, so maybe you should answer.
Can I just jump in, Fred Ross, before Ali comes in? I should have said, look, I think it's a no-brainer. You don't stop doing anything we're doing at the moment. Correct. So SMC has to continue. Bed nets have to continue. Longer lasting insecticides in bed nets have to continue. All of that is kind of priced in. What we're trying to do, there's still 500,000 children dying every year. What we're trying to do is add a new intervention. So when I talk about vaccines, I'm not saying use vaccines instead. That would be a step backwards. You want to use vaccines in addition to everything we're doing at the moment. Go ahead.
Excellent. Ali, please, go ahead. Ali, unfortunately, we can't hear you.
Yeah, thank you, Pedros, and thank you, Adrian. No, I think I do agree that as we are developing sort of this second generation vaccines, definitely it is important to ask ourselves how are these vaccines going to be used. And I mean, as the paper shows, that perhaps it would really depend also on the malaria epidemiology of the country. And I think the issues that Adrian talked about, sort of organization within the countries in terms of how different authorities are going to work together to effectively deploy the vaccine is something also that needs to be addressed. But I think, I mean, just to add, I think this is something that individual countries and researchers in these countries have to think about, and whether thinking of sort of a targeted approach, depending on the malaria epidemiology, like combination in areas of very high transmission with other means, sort of other intervention could be something to think about. Is worth considering. So I mean, I don't have an answer, but I think it is this could only be supported by I mean, both modeling, but also actually do some of the clinical field research to try and see how best these vaccines can be used in combination with other interventions in the field.
Yeah. No, thank you both. Thanks a lot. We're going to jump around a little bit, Adrian. Hopefully you do not mind that. Some of the questions will be fairly basic, but some of them will be more practical. Our next question really is, as someone who's worked in this space for a long time, and we were saying earlier how how great the landscape looks compared to the past. What do you think would be, in your consideration, what do you think are some of the greatest advances in recent years that would enable this forward acceleration in terms of different things, vaccine designs, testing protocols, whatever you want to talk about? What comes to the top of your list?
Yeah, I think the list you have is pretty good. Vaccine design has to be very near the top, or maybe at the top. Basically, if you don't have a vaccine that works, you're not going anywhere. So you've got to design better immunogens. The simplest lesson that we learned in malaria was in the mid-1990s with RTSS, which was the first nanoparticle or VLP, virus-like particle, whatever you want to call it, vaccine that worked. And looking back, it worked because it was a nanoparticle. And the soluble proteins, the peptides that had been tried beforehand, were simply not immunogenic enough. And annoyingly, if you look at studies in mice, that when we've done all of those studies in mice with various types of malaria vaccine, it's the same in mice. The clue was there. If you use a nanoparticle vaccine, you get higher antibody responses, and that's crucial. Sadly, in the late 1990s, the world misinterpreted what was going on with RTSS. The company sponsoring the vaccine and making it thought it was working because it was inducing CD8 T cells. We and others looked at that possibility. There aren't any CD8 T cells induced. Secondly, they thought that whatever way it was working because they had an amazing adjuvant. And those two observations are linked. They thought this amazing adjuvant, if it induces T cells and protects, is really, really important because we can use it for other vaccine types. Now, in fact, it doesn't induce CD8 T cells, as I've said, that are specific to the vaccine at least. And it isn't that fantastic an adjuvant. Yes, it's a good adjuvant. Saponin adjuvants are good. I have yet to be convinced that you need the TLR4 ligand in the AS01 adjuvant. If you take it out, very likely you would reduce reactogenicity. It isn't in matrix M. So we have a matrix M adjuvant without a TLR4 ligand that is as potent as one with it. So I think immunogen design is right at the top. Number two has always been a malaria strength. In other words, we can test vaccines for efficacy for $1 million, or certainly £1 million, in small numbers, because the human challenge model has been around for decades and has been really useful, and we wouldn't have 2 near licensed malaria vaccines if we didn't have the challenge model. I've talked about adjuvants. Yeah, I think they have been important. You're not going to get protection with R21 with an emulsion adjuvant. It just isn't as potent. Regulatory pathways have advanced enormously. So, if we moved faster with R21 than RTSS, remember we were following in the path of RTSS, which had paved the way for doing novel vaccine trials at scale in Africa, built up capacity in Africa, and we picked the best places to go, we thought, for our phase 2 and phase 3. And the regulatory capacity in 2019 in Africa is unrecognizably better than it was in, say, 1995. So that was a big advantage. Sorry, ETC?
Whatever. I mean, whatever. Ali, you want to add anything here?
No, okay, so if you may allow me, Pedros and Adrian, I wanted to just go back a little bit, and we were discussing earlier about the use, and maybe I thought this question could fit into that discussion. And the question was that, what do Adrian think about the vaccination of adults? So against malaria, and I'm talking, we know that there's been a lot of effort to target the children for obvious reasons. These are more, they bear the greatest burden of the disease, the mortality, and but with the changing in epidemiology and change in the risk over time, I think maybe he would agree with me, and that would become increasingly an important group to not ignore when we talk about malaria elimination. And I mean, the malaria vaccine haven't been performing pretty good in adults. So what do you think, Adrian, in terms of sort of moving forward, in terms of addressing this gap?
Yeah, as I hinted at earlier, I see this hugely important exercise of malaria control in two stages. The imperative in my mind is that we reduce childhood deaths very quickly by deploying vaccines in young children as widely as possible with suitable international donor support. And I think once these vaccines get rolled out and the impact is measured by people like you, Ali, and others on this call, donors are going to come in and say, look, we really can make a difference to malaria. Let's make sure every child is vaccinated and keep their antibody titers at a good level until they're 3, 4, or 5 years. I don't know what the right answer is in terms of how long we need to maintain that in young children to stop them dying, but it might be up till school age. The second stage is what I think you're alluding to, which is whole population vaccination and other interventions to knock down transmission and to cause elimination in, initially in countries with lower transmission and then eventually in every country in Africa. I think that is doable. I wouldn't have said that 5 years ago. But now that you've got vaccines that, at least in the short term, can give you nearly 80% protection with a single subunit component without mixing any components, yes, we should be able to do that. And we need to get on with it. And the vaccines should be as safe in adults as they are in children. We have preliminary data on that. Most vaccines start off in adults. They are not as immunogenic in adults as they are in children. That's probably true of most vaccines. So how would you go about this? I think you would start with an indication that is important, which is women of childbearing age planning to get pregnant. There's been a nice trial in West Africa, in Mali, by the Scenario group, showing they could get about 50% efficacy in that group with their intravenous sporozoite vaccine. So that's important. I think we could do better than that with a subunit vaccine in that target age group, and that would be probably an initial target for licensure fairly soon with R21. But after that, yes, we need to get serious about elimination campaigns. We need to answer the question, what happens when you stop eliminating? Do you go back to square one, as has happened before? And if not, how are you going to maintain that? And that's a large discussion. I would hope that by the time we are doing that, we have better vaccines than R21 that are 90-something% effective and last at least a year. And, I don't think you're going to get them last for 10 years. I don't think you're going to need boosters for that. But, what is the cost effectiveness of combining traditional malaria control measures and vaccination? And how are you going to stop parasites coming back the following year with mosquitoes?
And we're going to, I would love us to return to this question of combinations, immunogenicity and all that, different stages during our conversation today. And actually, we can start with you mentioned that these things are not as immunogenic as they should be. They are not as immunogenic in adults as they are in children.
Yeah.
In most cases. I mean, given this situation here, I mean, an important question, therefore, is just how well do we understand malaria-related immunity in humans? Do you think we understand it enough that we can go forward with this? And what are the major gaps? It appears that some of the things that we know happen in kids don't happen in adults. Some of the assumptions about immunogenicity vis-a-vis vaccine protective efficacies are not necessarily as previously thought. And we have a series of questions around this for you, Adrian and Ali. You could begin here. How well do we understand malaria immunity?
Yeah, let me give you a health warning in answering this. I'm going to upset some people, but I have to tell you this because it's true, and I'm absolutely convinced it's true.
Go ahead.
You don't need to understand natural immunity to malaria. You definitely don't need to understand it really well to make a vaccine. Look how well we understood natural immunity to COVID-19 in January of 2020. We knew nothing. We knew there were a few cases in China and some in Italy. A vaccine was designed. It worked. It was useful. Yet, if I got $10 for every review article that I read saying, what we really need to do to make a malaria vaccine is better understand natural immunity to malaria, I'd be a rich man. So look, let me explain it a different way.
Right.
Vaccines that are working against malaria at the moment, almost without exception, are producing unnatural immunity. If I produce the level of antibodies to CSP that you can find in somebody who has not been vaccinated in Africa, you will see no protection, because the levels were actually on the slide I showed in the control group. In those kids getting 3 episodes of malaria a year in Burkina Faso, their CS antibody titers are essentially 0. And they need thousands on that scale to be protected. So that is unnatural immunity. You never see it in the field. The same is true with blood stage vaccines. The leading blood stage vaccine, I would contend, is RH5. You can get some antibodies to RH5. They're really low level. To protect, you need much higher antibody levels, and they do protect. That's unnatural immunity. Nobody has those levels to RH5. Transmission blocking, you know about. There isn't much transmission blocking immunity. There's a little bit by natural exposure. Vaccines like PFS 230 do massively better. So, I'm not against understanding immunity to malaria. I'm just pointing out that it's actually a different field to vaccine design and development. So vaccines are the art of the possible. And we have better immunogens and better technologies now than we had 10 years ago. That's why vaccines are working.
Adrian, what does this mean then for when you're trying to identify, correlates to measure in a field study? I mean, you typically begin with a lab study, right? So what does your answer mean with regard to, field studies and how well they compare to laboratory studies? Does it mean then that we don't have to measure the same things?
I think if an immune response that protects against malaria individually against a single antigen, and remember, nobody's making 50 antigen vaccines against the whole parasite, guys, and that's a completely different field. You're not going to find out the best antigen unless there are protective immune responses to that antigen that strongly protect people against malaria in the field. And I don't know if there is such an antigen that's induced. What happens in the field is you have tens or more likely hundreds of antigens that contribute individually a small amount, and you acquire those immune responses, sadly often to red cell surface proteins that are variable both in sequence and in temporal expression. And cumulatively, that gives you useful immunity that by the time you're an adult is pretty protective.
So if you went out there and just measured one of these, you might observe very low levels and conclude that it's not working yet, actually.
Well, we have a lot of analytical techniques these days, if you were able to measure immune responses to every malaria antigen, and there are chips now and people are trying to do that, and you had all those documented, the problem is sample size. Because the more antigens you have, the larger the study you need, and the bigger the effect size you need to see it if you have multiple comparisons. So it's not impossible, but just go and have a look at the literature and look at the consistency of claims of correlates of protective immunity to major antigens. My reading of that is inconsistency is the rule, sadly. And it's not because the investigators were poor or did the study wrongly. They're just are very weak correlates that are hard to find. There must be some.
So 2 important messages. We do not need to fully understand the immunology of malaria for us to make a good vaccine.
Correct.
And number 2, there are a lot of inconsistencies in field measurements of these, correlates that we probably need a different way to observe them.
Yeah, and to be clear, that's not a criticism of the studies. The study is finding that you should do the studies just in case one antigen popped out and was strongly protective. That's why you do the studies. The reality is that doesn't appear to be the case. And that's a correct result. It's the correct answer.
Talk to us a little bit about this. When I read, I'm not a vaccinologist myself, but when I look at vaccine studies from my colleagues Ali here and Saidi Jongo and the rest, you always have these early studies where you describe immunogenicity, first of all, before you then go into the efficacy and all that. And for a long time, it appears that vaccine trialists always use this as a step before the efficacy trials, and the assumption that you need a reasonably high immunogenicity as an indicator that a candidate is going to be a good vaccine candidate. Now, some of your studies with your colleagues suggest otherwise.
I don't think. No, I don't.
Are we misinterpreting this? Go ahead.
No. So this is the work of Julius Hafala, who's very good. He's in London. What the title says is the low immune responses that you get kind of naturally to malaria pre-erythrocytic stages, and these are liver stages he's working on, can be overcome by vaccination. I completely agree. What he's saying is that natural immunity to the liver stage gives you modest immune responses that are weakly protective or not protective at all. And if you vaccinate them with a good subunit vaccine that gives you CD8 T cells, you overcome that modest immunogenicity and get protection. That's what the paper is saying. So I totally agree.
Yeah, so it means then that you shouldn't necessarily eliminate a candidate on the basis of immunogenicity.
Correct, in the sense that if you went down the road and started measuring children's anti-CSP antibody levels, if they've not been vaccinated, you'd see they're almost nil. You should not eliminate CSP because of that. And of course, you'd be wrong if you had, because you can make a CS-based vaccine.
Right.
Sure. Cool. And sorry, the other thing to remember is thinking about this parasite's been around for millions of years and has won, in the sense it's still around despite our best efforts. The reason it's been able to do that is that it's got lots of clever immune evasion strategies that has stopped our immune system getting rid of every malaria infection very rapidly. We get rid of a lot of them, probably most of them, very rapidly, but the parasite has evolved to continue to transmit.
Excellent. I mean, Adrian, I would like us to proceed to a different topic here. You and your team have always called for not only an effective vaccine, but a vaccine that is also deployable. So you have this concept of, the world needs a vaccine that is effective and deployable. And many things around that. So I would like to ask both yourself and Ali to describe that. But also, in addition, please do check the chat once in a while. And I would like to elevate a few of those questions for you at some point, because there are some very, very important questions in there that we should address. And I will be elevating a few of those. But first of all, describe this concept, both yourself and Ali. What do you guys mean when you say that the world needs not only an effective vaccine, but also a vaccine that is deployable? what is this deployability concept?
Okay, deployability is simply the ability to not just have a vaccine made at a manufacturing facility, but to have it usable in the real world. So what might stop it being deployable? The vaccine might be too expensive. If it's $300 a dose and you need 4 doses and you have millions of people to vaccinate, who's going to pay for that? I think the answer is nobody. It would not be cost effective. So low price, which implies efficient manufacturability, is the crucial starting point. Mm-hmm. Then you have to, within deployability, is the whole area of social science on whether people would take the vaccine. If people aren't taking the vaccine, it's not going to have impact. And then you have to understand why people might be hesitant or just anti-vaxxers and not want vaccines. So that's in there in the deployability concept as well. But mainly it relates to scheduling and dosing and the feasibility of administration of the vaccine. And feasibility is setting specific. So we know a lot about the settings where we most need a malaria vaccine, at least to reduce death, is in low-income rural areas of Africa, in West, Central, and East Africa. And, you know, the good news is that those are countries with some of the highest vaccination uptake rates in the world. You know, I tell my colleagues here that when they say, well, can you really deploy a vaccine in Africa against malaria? I say today in many African countries, 9 or 10 vaccines have higher uptake rates than you find in some parts of London, which is definitely true. So, you know, feasibility in terms of vaccine acceptance looks good. And I would add to that the important RTSS implementation trial or pilot implementation studies findings that in part address the question, if you gave RTSS at 5, 6, and 7 months of age, would mothers come? You know, they've already had their child pricked several times at 6, at 10, at 14 weeks, maybe another booster before then, and you want them to come back at 5, 6, and 7 months of age. Would they really do that? And that was asked by the WHO SAGE committee. And the answer is simply yes, they did in those 3 countries. And it looks like, you know, what I had hoped, that people, mothers particularly, were sufficiently aware that malaria is one of the most likely ways their child is going to die if they're still alive at 6 months. And a vaccine being offered should be welcome, and the uptake would be good. And indeed, it was.
Ali, please. Yeah.
No, I don't think I have anything to add there. I think Adrian has touched on everything in terms of deployability. I was also going to focus a little bit on the issue of communities that are receiving the vaccine, and I think it's important to also consider that when you're thinking about deployable malaria vaccine. And so this raises an important point that during the evaluation of this vaccine, I think some of the social science components should be integrated during the development. I mean, you don't want to have a very good vaccine, and then at the end of being ready to deploy, then you find, well, given these reasons, the community does not like it. So I think that sort of underscore the importance of when we do, especially sort of late stage vaccine development, sort of phase 3, some of these questions are embedded into the evaluation, into the trials, to be able to address these questions if there are any questions related to community acceptability of the candidate.
No, thank you so much, Ali, for that. And these questions are particularly important with regard to RTSS. I don't want to go back to the results of the RTSS vaccine, but of course, with the understanding that it's an imperfect vaccine, you still have issues such as, you need 4 doses of it. Of course, the pilot studies have demonstrated very well that they work effectively within the scheme, the other immunizations, integrated in the other immunization scheme, the EPI, for example. So it would be nice, just to think about what improvements can we make on deployability when it comes to what has been seen with RTSS. And by the way, there is a comment on the chat there about immunity, understanding blood stage immunity. Ali or Adrian, if you guys want to address that at some point, there's an argument that you still really need to understand that. But very, very briefly, we've seen the great results that came with the RTSS, demonstrated to work effectively well, and the WHO is recommending now, based on modeling work, that the best places to prioritize, would be the areas with the highest transmission intensity. So places with most of the malaria. This is work that was done in collaboration with many African scientists. Initial results, of course, also including a lot of African scientists in different sites, and then the modeling studies, and finally the WHO recommendations that did that. Adrian, here is a list of comments by yourself and by other people in early days before the pilot tests were done. And we just wanted to ask you whether some of these things have now been addressed. I mean, you had concerns about age, for example. Can you, what happens if this vaccine is not working for children younger than 5 months? What happens if you have to, deploy this vaccine many more times than that? Do you feel like some of these concerns that you had in the beginning have now been addressed, either after the pilot studies on RTSS or by the ARC-21 vaccine trials?
Yeah, sure. So the last one there is the easiest because we've just talked about it. The real problem arises if it's given at 6 to 12 months of age. There's the cost of delivery, which is increased. We know that because you have a different delivery system. We've talked about that earlier. And there's also the question of whether the children come back. And we now know that the mothers will bring their children back at 5, 6, and 7 months of age. So that's good. By the way, we have not solved with R21 the young infant problem. We don't have any data on R21 at 6, 10, and 14 weeks of age. We will be doing that in Mali very soon, but I would be very surprised as well as very happy if it worked really well at 6 weeks of age, because RTSS does not. The other issues, natural immunity. Yeah, this one won't go away. So you have to, with any new vaccine, accept that if you're preventing malaria, children will build up immunity to malaria, build up natural immunity more slowly. So a lot of work has been done with RTSS to look at that, particularly recently in Mali and Burkina Faso, and particularly on a 7-year follow-up of RTSS work published by Tinto and his colleagues. The short answer is it looks manageable. Your risk with RTSS did appear to go negative later on after you stopped vaccinating. In other words, at years 4, 5, 6, and 7, there was a hint in some groups of negative efficacy. On the other hand, with R21, we've seen nothing like that so far. We haven't followed out for as long, but it's something that we will continue to watch. We believe we're getting better immune responses with R21 than RTSS. That's not published yet. It's not even definite yet, but it certainly we have some evidence to that effect. That may help. We think we're getting better durability of immunity. Again, that's preliminary. If those turn out to be true, it should be less of a problem with R21 than RTSS. But this is definitely something we're going to be following. Our phase 3 trial, even though its primary endpoint is at 12 months, is not going to stop at 12 months. We will carry on following those children.
Right. My colleague Ali has investigated some of these questions about the immunity. Ali, what do you see since your early trials in Kilifi about the reversal of the risk? Do you think that this is a problem that has been solved now, or are we still at the risk of the children who are vaccinated becoming, experiencing greater risk than those who have not been vaccinated after in the studies that you did, this was observed after 7 years. But what are your thoughts now?
No, I think yes. I think Adrian has alluded to it. I think yes. It's what we showed in that phase 2 study is that 3 doses, and children were given 3 doses. Didn't give the booster. That's the important thing to note. So 3 doses later on in the sort of the follow-up years, there was a negative efficacy that offset what sort of gain had been sort of seen early on during the follow-up. So this, of course, was a concern. We published on this, and I think sort of phase 3 went to sort of test the booster dose, which is very important. And in the result, of course, the booster dose was very critical to sort of delay this this age shift, if I may call it. So I think the point which has been raised there in your slides, Pedros, I think is still important to look at it critically, especially in the real setting. If the fourth dose of booster becomes problematic and less mothers do not show up, then it could be a challenge in a context where people are very far away. The access to healthcare is a challenge, etc. But if the fourth booster can be, most mothers sort of go for the fourth booster dose, then maybe it becomes less and less of a problem. But I must say, managing malaria is it's not about only giving the vaccine and forgetting about the whole healthcare system issue. So I think we have to look at it in that context. So it may be a problem if, of course, there's a problem with the fourth booster, and then poor healthcare systems where people cannot go to health facilities and get care. But if we have a very good number of children coming for their fourth dose, maybe that is manageable. But I want to make sure that this is very clear. We didn't look at the fourth booster dose, and the fourth booster dose is very critical, at least from the phase 3 study. And I understand that I don't know if Adrian knows if there's any updates, but I think the fourth dose was still being evaluated. I think the results that led to the recommendation were purely based on the first 3 doses. And I think from that report, the fourth booster, the feasibility and the impact of the fourth those booster was still being evaluated. So maybe we'll have to wait and see what did come from the field over.
Right. Yeah. The WHO recommendation has a fourth dose, I believe. Adrian, this correct?
Yeah, that's right. And just to say there are further data coming. So in the phase 2B in Burkina Faso, that I showed you some data from at the start, we gave a fourth dose, but then when the time came for a fifth dose, we randomized children into getting a fifth dose or not a fifth dose. And then a year later, children have just been randomized last month to getting a sixth dose or not a sixth dose. So we should have children with 4, 5, and 6 doses being followed as we go forward to see if any extra doses make a difference.
Excellent. Let us wade a little bit into chemo prevention and then back to vaccination. So here is some excellent results from our colleagues again in West Africa demonstrating the power of seasonal chemo prevention. And we are hoping that we are going to have a masterclass soon on chemo prevention broadly, and hopefully we will have Professor Ndiaye joining us among others to discuss some of these topics, and also a Tanzania-specific masterclass on chemo prevention where they will talk about some of their early results. But after this trial, one very interesting thing that has happened is that we have had our colleagues led by Brian Greenwood and Daniel design these studies where they combined RTSS with SMC. These results were published last year, fantastic results here as well. And you see very, very clearly that putting together RTSS with or without chemo prevention gives you very different results. If if you put these things together, you have a much, much higher protection than either vaccination alone or chemoprevention alone. And if you just see here, you see some of those additional benefits go up to 60-70% increased protection. Now, back to you, Adrian and Ali. When you talk about deployability here, this is a case where you have kids, you're following them every month, and in addition, you're giving another 3 doses of the RTSS. Of course, you're getting a much better value and protection of there. So talk to us a little bit about, and I've seen a paper from your group, Adrian, a commentary where you guys describe this as one of the best successes in recent years. We'd like to hear from you a little bit about, the value of this as something great, vis-a-vis the practicality. Seasonal chemoprevention plus RTSS, in both cases more than 3 doses. Of course, fantastic results. But how do we exactly do this? And should recommendations be aligned to such a combination? Back to you, Adrian.
Yeah, so the answer is different for West and East Africa, isn't it? In much of West Africa, not all of it, there is seasonality of malaria. And in the settings that this trial was done in, there's striking seasonality. As I said earlier, 75% of the episodes in 3 or 4 months in the year. Now, if you're a vaccine developer, you don't have a choice about SMC. SMC is policy in those countries where we were giving the vaccine. So in our phase 2, the government provided, National Malaria Control Program provided SMC. Not every child got it. We recorded who did and who didn't, or at least the field workers did, so we have those data. Our data are the same as this paper in the sense that if you give one intervention that's SMC, that works fairly well, and you give another intervention that's a pre-erythrocytic vaccine, if you give the 2 together, it's the sum of the parts. It's not synergistic, it's additive. But additivity is a lot, as you can't see from this trial because there wasn't any group not given chemoprevention. We had the advantage of some children who just didn't get chemoprevention because the difficulty with SMC is getting it to every child in the real world. So yes, I mean, I don't think we have a difficult decision to make here. SMC should continue. We should increase efforts to make sure that in routine deployment, as distinct from an excellent clinical trial, as done in this New England Journal of Medicine paper, that nearly all the children get SMC. And then the vaccine will still work, whatever it is, 75, 80% on top of the SMC, which is what this paper is showing, which is great and makes sense, of course. So I think that's not controversial, and SMC is cost effective. Interesting to see that RTSS was slightly more effective than perfect SMC here, and we would anticipate R21 would be better still. But that leaves East Africa, where there's less seasonality, and SMC may not be policy as it isn't in many areas. Should you start doing that for areas with less intense seasonality? Start doing it anyway. And then you get back to an old debate about, potential disadvantages of drug chemotherapy for all children. Will you increase drug resistance? Is it cost effective, et cetera, et cetera. So, we were hoping that vaccines to some extent would help overcome that, because looking at the literature, and clearly I'm a vaccine guy, but if you look at the efficacy of R21, you look at the efficacy of impregnated vet nets, you look at the efficacy of RTSS, you look at the efficacy of spraying, you look at the efficacy of SMC in real world deployment, none of them hit over 75% as a single intervention. No. So that's why we're excited about a good pre-erythrocytic vaccine coming along.
So what you're saying here is that in the event that a country were to adopt, so let's talk West Africa first, and then I would like Ali to talk East Africa as well. So in the West Africa situation, or in the Sahel, What you're saying is that in the event that they decided to use RTSS, the recommendation should be to always use it on top of SMC and never as a replacement of SMC.
Not only is it, that decision has been taken. So WHO's plan is very, very clear. You carry on with all existing interventions, don't stop any of them, whether it's bed nets or spraying or SMC, and you add a vaccine on top of that.
Excellent. Ali, how does this play out in East Africa? Of course, with the understanding that at the moment we do not have any medication, we cannot use amodiaquine in the south of the Sahel. So East African situation is not necessarily a good place for SMC. I know that there are some trials ongoing, but how does the RTSS SMC combination, how would that play out theoretically in the East African setting?
Yeah, I think that's a challenge because, as you said, the epidemiology is slightly different. I know the Ministry of Health, in collaboration with NIMBRI, are working to sort of, because the country has been stratified very well, and there are only some sort of few areas in the country where you do see this seasonality sort of similar to what you might be seeing in the Sahel region. But I think I'm just looking at as a proportion of the country, or the proportion of the sort of a problem we have, and whether this will be something cost effective, and that there are no any other approaches that could actually work. I see a challenge there. I think, we may end up sort of taking the advice of the WHO that, they've recommended this, at least RTSS, for areas of high to moderate transmission. And obviously, we could target this area. I've just seen a question from the chat, maybe also Adrian might want to answer it, is that, with stratification and targeted sort of intervention, there could be some other issues. There could be some community backlash in terms of, how do you decide where to go to vaccinate this particular group of, this particular area versus this area. So I think there definitely we need in East Africa as a country to look into the vaccines that are being produced, look at our epidemiology, and determine how best we can use these vaccines. Bearing in mind there are a region in Tanzania where people are not very much exposed, and moving within the country may predispose some people as well. This is something people should also remember. Tanzania is sort of one country. If I live in, in the slopes of Mount Kilimanjaro most of my time, and suddenly I take a trip to let me guess, in Kuranga, for instance, I may get malaria, and someone could suffer from that. So I think we need to think very carefully as a country from the stratification, how this vaccine is going to be used. So I don't have an answer, but I think it's an exercise that a national malaria control program and other researchers need to come together and figure out how best to do it.
The question on the chat was actually asked by a member of the Tanzania NMCP. So why don't I invite Dr. Lushashi, if you are still on the call? Are you there?
Yeah.
I saw you raise your hand. So do you want to talk a little bit about how the vaccine would play into the landscape given the stratification problems in Tanzania? Dr. Lushashi, come on, please. I hope he is still there.
Hey, hello. Yeah,
hello,
Pedros.
please go ahead. Yes.
Oh, thank you very much for a very good presentation. My question was that we have a stratification in our country, and we are still learning about the vaccine. So we don't know maybe how to go about it. The area, according to the stratification, are very closely between very low transmission and high transmission. Across the regional district and even the wards. So I don't know the experience behind it. How gonna we manage it if it happen that the vaccine now is conducted in our country? Thank you.
Thank you so much. Thank you. Adrian, Ali, you guys know try and respond this.
Ali, why don't you start?
Yeah, I think it's a difficult question. I don't think I have all the answers, but I think we might have to think about again vaccinating based on our stratification and targeted the areas where we think the vaccine is going to have a more significant impact. But I also think we need to consider, as I said earlier, that the political ramification, the community backlash, the perception of the people so there has to be a very, very good maybe something to underline is a communication and involvement of the public at a very early stage, whatever the approach we're going to take, so that this is very, very clear what strategy is taken and why. I think involving the community and communication will be very, very important, I think. And then determine where best the vaccine can work, where we can reap the benefits of the vaccine. That will be something else. So giving it based on certification might be one approach, but also looking at the epidemiology and see how else can we use it in combination with other interventions in areas where probably we do have this seasonal transmission, something as we see in Mali, actually in Sahel region. So that is what I can say for now, but that's definitely something which needs to be discussed.
No, definitely, yeah. Adrian, please. And Adrian, I know that the question is mostly in line with RTSS, but we have the R21 results starting to come, so mix it in if you wish.
Yeah, well, the question really is for RTSS. What is really frustrating from the point of view of the team here is that there's a massive discussion about how you make 7 million doses, let's say, fit into a need for 160 million doses. Why should a malaria vaccine be rationed by a manufacturer when COVID vaccines have been, everyone's been killing themselves to make as much vaccine as possible, and that's succeeded.
There's been an oversupply, if you like.
Yeah, which is a great problem to have because most of these can be frozen and can be kept. 12 billion doses in a year. The record was 500 million, less than that in a year for any disease. So, this is an RTSS problem, not an R21 problem. Serum Institute have said they can make 200 million doses. They can make more if necessary, and they will once elimination vaccination starts. And remember, a lot of that's going to be in Asia, not in Africa, low transmission areas. So hopefully we're trying to solve this rationing problem by getting this vaccine R21 licensed next year and manufacturing, as Serum did for COVID, at risk before it's licensed, so that as soon as people are ready to deploy, the vaccine is there and the vaccine supply won't be rate limiting. What I see as a much bigger problem is funding for the implementation. Who's going to give it? Is it EPI? Is it the National Malaria Program? How is it going to be given? How quickly can people start? Because the sooner you start, the more lives you save. And believe me, we've been waiting for decades for a malaria vaccine to be rolled out. Let's not waste another 2 or 3 years after it's available by fooling around trying to organize it. We're giving notice now. These vaccines are coming. There's going to be enough of them between the two of them. And the challenge then is to use it as efficiently as possible to save lives. And you don't have to think too hard about doing other things. You're doing those already. Just bring the vaccine in on top and see what the impact is. Definitely observe impact. That needs to be measured in some way. And most countries are relatively good at doing that.
Excellent. We have I hope you won't mind, because we're going to bring this question back about the limitations in vaccine doses for RTSS, the manufacturing concerns, and how different this will be between RTSS and R21. I hope that for Dr. Lushashi, that this is, and also to other members of NFCPs across Africa, that there is a greater understanding that in addition to the stratification questions, there are also concerns about the availability of the doses. The fact that we have so little RTSS vaccine available at a time when people are over manufacturing, if you like, the COVID-19 vaccine. So I think there you have a more of a political economy question.
Fedros, does everybody understand on the call why the supply of RTSS is so limited? Because I suspect some people are
we assume that not everybody understands, so we actually have a slide on that.
Okay, right. I think it's important to come to that because you might be able to tweak that a bit. Yeah.
Correct. Yeah. So we will ask you a question about that. Before we go to that, I would like us to run, over the next 4 or 5 slides, very quickly about, the WHO targets, the 75% roadmap goal, 75% prevention against cases and 50% prevention against severity. Just clarification on whether this is still, the target. Why is it the target, or has it changed? One of the biggest stories when the R21 vaccine results came out was that it was the first one to beat the 75% WHO target. So that's something that I would like us to talk about very briefly. Why, and this is a question we asked also our Sonaria colleagues a while back, what is it with the 75%? What is the justification here? And do we foresee a situation where this would be higher or lower? Back to you,
Adrian. So it's an arbitrary number. I was there in the room with many other people when that number was agreed. Some people wanted a lower number, saying, come on, let's be realistic. We'll be happy with 50%. Why 75%? And other people said, no, 90% would be better than 75%. So the consensus was 75%. It's not a magic number, but clearly the more, the better. What looks odd now, this is 2013, is that 75 sitting beside 50%. There's no logical reason why efficacy should be lower for severe malaria than for clinical malaria. You can make an argument the other way around, that you might do better, as you do in many vaccinations, against severe disease than against mild disease. You understand that for COVID. The vaccines are much more effective against severe COVID than against clinical COVID. So I think that's just wrong. If you prevent 75% of cases, your best estimate of how many deaths you would prevent from severe cases is also 75%. That's
is this something that we are learning only after COVID, or is something that was just
no
flu vaccine missed in the beginning?
No. Flu vaccines do the same, etc., etc. Most vaccines, and you could think about the biology. What's happening? The immune response is fighting against the pathogen to try and control it and reduce. It's easier to control it a bit than to eradicate it completely. So yes, that's well known and happens with most other vaccines.
Cool. Wait, did I interrupt you?
No, I was waiting. You changed the slide. I was waiting for you to say something.
Yeah, I mean, a lot of people are really excited to go back into the R21 vaccine and ask some key questions here.
Mm-hmm.
But let's begin from where you left when you presented in the beginning. RTSS and R21 are in many ways the same. In some ways, they're different. So it is not like an entirely new vaccine. It is some kind of a variation of RTSS. And in my opinion, one of your greatest papers, some people might say otherwise, is actually this published in not necessarily the best journal, but a fairly good journal. We think that in terms of the history of R21 is probably the most powerful of this. Talk to us a little bit. I know you described this in the beginning. How different and how the same are RTSS and R21?
Yeah, well, they're the same in that they both use the circumsporazide repeat as the key component. Some people see that as a great similarity. I don't. I would remind you that every COVID vaccine that has been licensed has the same spike antigen in it. People will have duels about which is the best COVID vaccine and feel very strongly that one is better than the other. In fact, they are all much more similar than R21 and RTSS is, because they're all producing antibodies against the spike protein at fairly high levels. I'm sure they're not as high as with either RTSS or R21. So yeah, let's not argue about how different they are or how similar they are. They are different. And the most important difference is probably in manufacturability, as we emphasized. That's why you can have more R21 at a lower price. But the design here is not trivial because, remember, about 40 different CSP vaccines have gone into the clinic.
Right.
Two of them have reached 50% efficacy. The rest didn't. So it's not just the antigen, it's how you design it. And today, nobody will be surprised that a nanoparticle or a virus-like particle gives higher antibody responses. But believe me, for most of this century, people didn't accept that as being the key maneuver. So what we wanted to do was to make a VLP like RTSS, but to avoid the really strong hepatitis B immune response. So if you look at RTSS, you're getting competition for antibody induction between the NAMP there and the 5 times more abundant hepatitis B there. And you already know that hepatitis B is a very good vaccine. It was the first subunit vaccine ever licensed for human beings. It's pretty immunogenic. And you've got 5 times as much of it as you have of the malaria sequence. So we thought that'd be the easiest thing to try and correct. And I even spoke to Joe Cohen, who invented RTSS way back in the 1980s. And I think I annoyed him by saying, didn't you think of removing the hepatitis B? We thought we'd try that. And he just looked soulfully at me and said, good luck. So I interpreted that to mean that he had tried very, very hard to do that. Right. And of course, it wasn't possible because we tried to do it and we couldn't do it for a long while. We were working on an improved RTSS from about 2000, and it was 2012 by the time we had R21 working even in a mouse. So this paper took a little while to produce. But the key point I want to make is if you vaccinate a mouse with R21 and you measure immune responses to malaria, they're high, as with RTSS, but the antibody responses to hepatitis B surface antigen are very much lower. There's your positive control, there's the antibody. There isn't much antibody to hepatitis B after you vaccinate with R21. Right. So to an extent, we've overcome the problem we were trying to solve, stopping the RTSS vaccine being a fantastic Hep B vaccine, which it is, and try and make it a better malaria vaccine. And that was the project that Cass Collins, who wrote this paper and worked on this for her DPhil, cracked really. So that was excellent. Quite an important.
So in summary, could we then just say that by removing that second arm of the hepatitis, you are able proportionately to express more of the CSP?
You can say you get a much higher ratio of malaria antibodies than hepatitis B antibodies, where it appears to be the opposite, more hepatitis B with RTSS. And that's not surprising. That's generally agreed. Yeah.
Excellent. And you did describe these results already in your introductory remarks. I just want to highlight them one more time here. These are trials from West Africa. Initially, when this was published, it was only 6 months of data. It shows here very well that you have 70+%, 77% protection at best when given to children using matrix M adjuvant. We will talk briefly about that. Adrian, one of the criticisms about this paper has been that actually, if you look at what RTSS had in the first 6 months after rainy season, that you see the same results. What are your thoughts about this criticism?
Yeah, well, that's a rather arbitrary length. If you look at the first 12 months, it's 44%, and I showed you that on the slide earlier. So if you want a 6-month vaccine, yes, RTSS is pretty good. If you want a 12-month vaccine, it's clearly less good in that population. If you want it to be durable for 3, 4, or 5 years, then you want higher antibody responses later on. And I think we'll soon be able to report on that with R21 compared to RTSS. But to be clear, we're not competing with RTSS. We're improving it, and we're providing more of it. We want as much vaccine out there as possible next year and in the years after that. And RTSS is part of that solution. And the more vaccine, the better.
That's an important point to make, Adrian. That the idea here is not to compete RTSS, but to improve it. And we put up a slide there, if you like, that you could use to kind of just illustrate or summarize what the additional gains are here. Of course, I'd like to ask.
You've written at least 50 words on that supply and left out the most important one.
Which is the manufacturing? Is
the word supply, SUPPLY. If you can't get the vaccine, it won't work, I promise you. So that is the biggest difference in terms of real world impact.
Let's talk about that then really quickly. The supply part. Here's a report from Tony Blair Institute, also based out of here in London. They have looked at vaccine manufacturing capabilities in African countries. Very, very little, and except from their report suggests that 17% of the world is African, but only 1% of the vaccine is produced in Africa.
Yeah, which is dreadful and needs correcting, as we've seen. Yeah. Mm-hmm.
They've also demonstrated, however, that the market is quite huge, and that there are some countries that have started. You have, for example, Senegal that is already exporting GLP level vaccines of some kinds. And many countries are getting towards that. Now, another thing that has happened recently is, you have this publication from Reuters that suggests that the world's malaria vaccine might not reach millions. And this is, despite the fact that Gavi has just opened its doors for applications. And you talked briefly about this earlier, as talking about the limitations, the limited doses that are available from GSK and the manufacturer and so on. Now, in your publication, in the 2021 publication, one statement that you make there, and I guess this is what you wanted us to present on this slide, is that an important additional advantage of the R21 vaccine is the potential for large-scale manufacturing, low-cost supply to support global efforts. Now, I know that you guys are very particular about this specific attribute. Very simple terms. Please explain again. I know you've talked about it several times. Why is RTSS easier to manufacture? And how does this solve the problem that we currently have, that WHO has an approved vaccine, Gavi has allowed countries to apply, but if you ask the manufacturers, they are unable to provide even 15 million doses every year until maybe 2024. So what does it mean when you say that R21 solves this problem?
Yeah, okay, well, let's just be really clear about what's been approved. RTSS has been approved for wider use by the WHO, subject to pre-qualification or to regulatory approval. So WHO doesn't give national regulatory approval. Countries in Africa have to do that. The countries are of course, sovereign. So we're waiting, at least I haven't seen a pre-qualification approval for RTSS, and that's the regulatory part. What a policy recommendation says is we've seen the data on using this vaccine and its efficacy and safety, and if that meets regulatory requirements, we're giving a recommendation that the policy should be to use it more widely. But there's an if in that sentence. It needs pre-qualification. So it's not approved yet. And I'd like to have $10 for all the publications I've seen saying RTSS is approved for use in Africa. I think it will be soon, but it hasn't been yet, to my knowledge. Please correct me if I'm wrong. I haven't seen the pre-qualification announcement. So why is R21 better is the question you're asking, and why is there so much more of it? And there are many factors. Some of them are improvement in the yeast strain that is being used. It's Pichia that's used by the Serum Institute of India to make its hepatitis B vaccine. And you may know how expensive that is. It's cents, not dollars, for a dose. So manufacturing yield has been improved over the last 25 years. But, you know, the main problem with supply of RTSS is not the supply of RTSS. It's the supply of the AS01 adjuvant. And the two go together, and you need the adjuvant to make RTSS work. So what's the problem with the adjuvant? It's a pretty good adjuvant. It has three components in it that are well known. The MPL, which is manufactured, I think, in the Midwest of the United States. The saponin component, which is QS21, which I understand is manufactured in Massachusetts, and the liposomal formulation, which is added in Belgium. So you have three manufacturing locations to get a GMP lot of AS01. That's not actually the problem, even though it does make it a bit more expensive, I guess, doing it that way. The fundamental problem is getting the saponin component, QS21. Where do you get that? From the bark of a tree. And the real problem is that that tree only grows in Chile, in South America. So it's getting enough Chilean trees to take the bark from, to extract the saponin, to run the saponin on an HPLC column, to separate it out into fractions, and take fraction number 21, and make sure that's pure enough and all the other tests, and mix that into the adjuvant as one of the adjuvant components that is needed to make the adjuvant make strong antibodies when you give it with RTSS. Now, that's not a news. We've known for years that that's limiting. So if the criticism I've heard of GSK is not that they haven't got enough adjuvant, but that they've known for 20 years that a malaria vaccine is coming along. They know how many people there are in Africa and how many children. How come we're in a situation where there are only doses for 5 to 15 million doses a year? And I don't know the answer to that, but that's the question, you know, you'd like to ask GSK. I think part of the answer is that it's not the only vaccine that needs AS01. There's a shingles vaccine that's highly efficacious and marketed in North America and in Europe and sells very well for people of my age who would like a shingles vaccine, and AS01 is needed for that. You may have seen in the last month or two that there are exciting data for a childhood vaccine called RSV, which is also given to older adults from GSK that needs AS01. Now, that vaccine is not marketed, not approved yet, but it's coming and it's likely to come very soon. I'm worried that's going to be an even greater call, potentially, on doses of AS01. So maybe there's a magic solution in the pipeline, but you know, we've been waiting for 15 years to see it appear to increase the supply of AS01, and I understand GSK are trying, but you know, I think if there's an improvement, it might go to maybe, I'm guessing, 20 million doses, but I can't see it going to 300 million doses, which is what you'd really like to have. So it's not that there's an easy problem to solve. There's a really, really, really tough problem that the Gates Foundation have tried to solve and invested in that area to increase QS21 supply. But it's, you know, it's not a quick fix as far as I can see.
How does R21 solve?
How does it solve it?
It's a different adjuvant, right?
It's a different adjuvant, and it's a good question because
you want to talk about that?
Yeah, I'll talk about that. It's the same bark.
Just put up a little slide for you there.
Yeah, same bark, same tree, and you get the saponin out, and you fractionate it again on an HPLC column. Now, remember, there's no monophospholipid A, there's no liposome in there, so it's just the bark of the tree and that component fractionated into fraction A and fraction C, and they simply mix those together. So why is it kind of the same adjuvant? Because part of fraction C includes some QS21. And that's the adjuvant that Novavax told the world they would supply for COVID at a scale of 1 billion doses a year, or I think maybe more. And that is because they have a very efficient process for producing a lot of matrix M. Now, it turns out that their COVID vaccine was delayed in its approval, certainly in North America. It now is approved there, but they aren't supplying 1 billion doses this year. But what encourages me is that for malaria, if we had 100 million doses, that would be good. 200 million doses is what serums say they can manufacture, and they can manufacture it too, I understand. So that there's less of a problem because you're using far less of each gram of bark to make a dose of matrix M than you are of QS21. Hope that makes sense. And it's far simpler and less expensive to do so.
So shall we start growing this tree? Shall we start having plantations of this in Africa?
Well, let me put it this way. If you do, you might become a very rich man.
Yeah.
Sorry, the bad news is it doesn't appear to grow in any other countries that have been tried, or even in greenhouses. But there are long articles in US magazines that you can read about this. It's not easy.
Ali, looks like there's a path to wealth, to riches here. If you and me can, I mean, we grow a lot of chick in Tanzania. We could. It's amazing that I realize that most people do not, and myself, I didn't understand that the adjuvant for the RTSS is a botanical, and that it's the limitation on the botanical side of it that makes manufacturing for RTSS such a difficult problem. It is great that the matrix M adjuvant production process slightly improves on this, and hopefully we can get a greater access with this, given also the results that have been shown. I would like to recognize the arrival of my colleague, our other co-host, Dr. Sheila Ogoma. When we started, Sheila was on a flight to Cambodia, as usual, doing our malaria work, supporting countries. She has arrived and has joined us, and I would like to ask Sheila to turn on our video as well, and in case she has any questions for that. Ali had some question for you as well, Adrian. We would like to go back to that before we proceed to the next. Ali, please.
Yeah, just a question. I think I wanted to ask when we were discussing about the, what R21 is doing is sort of improving on RTSS. So I wanted to ask Adrian about the whether there is any evidence from the immunological point of view of the improvement of the antibody responses that we see when we are vaccinating with R21. I mean, could you talk about something about that?
Yeah, so that's the study we've wanted to do for at least 5 years, maybe 10 years. And to somebody like you, it will seem a very simple thing to do because you've vaccinated so many people with RTSS and you've now vaccinated so many people with R21. Why haven't the antibodies been compared? And I think you know the answer. We're not allowed access to GSK's sera from those trials. And believe me, we've tried. We are trying to work on a workaround as we speak, because I think it's in the public interest to be able to answer the question that you have just asked. But remember, your question is a subset of perhaps the most important question in all of vaccinology. Why don't regulators insist on comparative immunogenicity? They don't. Where is there a regulator who said, we've got 40 COVID vaccines, we want somebody to show what their immunogenicity is using a standardized assay, so we as regulators can know which is the more immunogenic vaccine? They don't do that. Everybody uses their own assay and uses a different y-axis with arbitrary units, and we don't know what the immunogenicity is. Let me just say that indirectly comparing and inferring various ways, we think that certainly the Group 2 R21 matrix M formulation is giving higher antibody responses, but those are still confidential data that we can't share because of that implied comparison to RTSS. But if somebody who has tested both R21 and RTSS wishes to do that in their own lab, I think that might be a solution. Be really pleased to see that.
That's interesting. Ali, do you have a follow-up question?
No, I mean, I think I don't have a follow-up. I think not follow-up question. I think it's something to ponder around and yeah.
Excellent. I guess in the field when you're working with colleagues and the collaborations in the malaria community as well. I guess the point Adrian just made last. You want to do things, but you also want to build partnerships, and so sometimes the things that you do might appear to be intentionally contradicting the others, and you don't want to do that. And I think personally, I'm very encouraged by what we have seen in the field. I mean, when you talk to our colleagues in Sanaria, they are very appreciative of the work that is being done at Jena, and when you talk to Adrian, he is very appreciative of the work that the RTSS team done. And if you look at their histories, seems like everybody has started from working on RTSS. So. And that it's a question of how do we improve what we have rather than how do we necessarily make a better one. I realize that in the media, they have occasionally referred to Adrian's group as developing a rival vaccine. And the term rival vaccine is probably not the most appropriate. Really, it's about how do we improve on what we have. Now, there are some questions on the chart here about the mRNA vaccines, Adrian. We are also aware of the work that is happening at Sanaria with the sporozoites, whole sporozoite vaccines, especially their concept of CVAC. How do you attenuate not with irradiation, but with malaria medicine? And now we have the R21 and we have the RTSS. So this portfolio is kind of expanding. The WHO roadmap has the slide where we're showing the WHO roadmap, we also showed a little portfolio slide there. So it's an expanding portfolio. My colleague Ali here in Bagamoyo is working on RH5 and many of these. Very broadly and briefly, can you talk to us about which of these opportunities or candidates is the most exciting and why? And of course, here we're asking you to put R21 on the side. Just talk about these other candidates. When you look at the entire landscape, which of these candidates is most exciting? Some of them are not even candidates yet. Our Moderna colleagues just say they want to start mRNA, and everybody, went very happily wild about this. So when you look at all these stories, which is the most exciting and why?
Yeah, so just remember when I answer this that I do come from Oxford University that has multiple other vaccine candidates for malaria in clinical trials, some of them in Africa. Let's start with RNA that you said drove people wild with excitement. I would be excited if you could show me an immune response to an mRNA vaccine that is as high in micrograms per mil as we can get with R21 in matrix M. In other words, in an objective measure on the same scale, of course, there are different antigens, but in micrograms per mil of antibody that's specific for the antigen, you can do that comparison. It hasn't been done. So all we have to do, again, is guess from indirect evidence about how strong the antibody responses to mRNA vaccines are. And, they're not hugely different to viral vectors. They're a bit higher to start with, but the overall efficacy of a chimpanzee adenovirus vectored vaccine against COVID and mRNA vaccines is essentially the same. There are lots of papers showing that. The antibody titers may be higher to start with, but of course the viral vectors produce more T cells. So, that doesn't get me excited about mRNA for malaria if the antibodies are only about as protected as you get with a viral vectored vaccine, because we've done lots of viral vectored vaccine trials for malaria and don't get very high efficacy as we do with R21. The other thing that discourages me a bit is that in data presented at TropMed last year, the Walter Reed lab in the USA had vaccinated mice with mRNA, I think working with a very good academic group as collaborators. And the efficacy in mice was, I think 60%-ish, maybe 70%, but nothing to write home about. And certainly not obviously better than other subunit vaccines. So, I think one has to really ask the question, what is it about RNA that would want you to do it? Is it faster manufacture? Well, that's not terribly important for malaria. If we're going to produce hundreds of millions of doses a year, this is not a key determinant that you could manufacture faster. And then you have the well-known disadvantages, higher cost, a $25 vaccine for COVID compared to a viral vector at a few dollars, and R21 at the same price. And most importantly, of course, thermostability. Are we really going to be using -70 freezers to store a malaria vaccine in the coming years? Now, I'm an optimist. I think the thermostability problem can be solved with mRNA vaccines, but it is not solved yet. And I'm not confident that Moderna or Pfizer or BioNTech are going to prioritize malaria and do the amount of work that's required to get a malaria vaccine all the way, into field trials and large-scale phase 3 trials, et cetera, because those companies often behave as if they really are profit-driven, and there isn't profit significantly in malaria. So by all means, we should be looking at mRNA vaccines, but there are better candidates out there. I would include RH5 on that list as a blood stage candidate that has shown efficacy in humans in a challenge trial, and that is published. The whole sporozoite vaccines are making progress. They've been coming for some time. Scenario was founded in 2002, but they're a very dynamic program. Lots of people who know lots and lots about malaria and pushing forward. And their challenge, of course, is cost of goods and route of delivery. And many of you know more about those vaccines than I do. And hopefully they will come along eventually as well.
Sorry. These are, especially with regard to the mRNA vaccine questions, are questions that have dominated and most likely going to dominate the conversation for a while. And sometimes we started to think, are we creating an illusion here that it's all of a sudden easy to make vaccines? Is it possible that companies like Moderna are underestimating what it takes to do this for a parasitic infection? Could the success with COVID be creating this a false, or an illusion that actually now all of a sudden within a year or two we will have a malaria vaccine?
Yeah. Oh, okay. One by one. I don't believe Moderna are serious about malaria. I think BioNTech are a little bit more serious. I think Ugur Sahin finds it fascinating, but he has told everyone, if we were listening, that it's going to be extremely difficult.
Right.
So, this is a guy who can make cancer vaccines. He thinks malaria is really difficult with mRNA. So I think that's probably true. I think that the big decision for Africa is whether mRNA is going to be useful for more than COVID vaccines. It's been tried for flu. Our flu vaccines aren't very good, 50-60% effective in a good year. The reactogenicity in the first trial for flu didn't look great. Nobody is out there trying to replace childhood vaccines with mRNA. Why, if some people think it's better? I think people who know about vaccines are a lot more realistic than some of the publicity. Remember, we had journalists writing about COVID who didn't know anything about vaccines, and all they knew was that mRNA vaccines were good for COVID. Well, hang on a minute. Probably the best vaccine for COVID is quite possibly Novavax's protein in adjuvant vaccine. It just took a bit longer to manufacture. Now, we don't know that, but it's right up there with the mRNA vaccines. And to an extent, that's the technology we're using for malaria. Only with malaria, we have a real nanoparticle, whereas the COVID vaccine from Novavax isn't quite a nanoparticle. It's structured, but not spherical. So, you know, I suggest that you not take your advice from the popular press on what's a good vaccine, but talk to people who've been making vaccines for a little while, and even listen to the head of BioNTech when he tells you that malaria is going to be really tough with mRNA vaccines, because malaria is really tough with anything.
No, thank you so much, Adrian. I would like us to, while we are on this, I would like us to just take up, 1 minute. I would like to make a quick announcement or correction here. So one of the WHO staff who is in the audience would like to make a minor correction, but an important one, with regard to a statement that Professor Adrian made earlier, which is that, and I think this is slightly unusual, but it's an important point to make. And I would like to read the statement as such, anonymously. I would like the statement here it goes. Number 1, dear Fredros, I would like to correct the statement regarding RTSS. On July 15th, July 15th, WHO issued a pre-qualification approval for RTSS. It is not usual practice to announce this proactively, but the product is now included on the list of pre-qualified vaccines on the WHO website. And we're going to put here a link to the website, just for those who would like to see that. So this is obviously not the normal path that these products take, but it's important that we get that clarification from WHO. We are also informed that if anybody has any additional questions regarding the pre-qualification of RTSS, that they could direct these questions directly to WHO, and I would be happy to act as a mediator to help direct these questions to the persons concerned. I'll put that on the chat, but I would also like to mention that our colleague who is sharing this information is not authorized to speak on behalf of WHO. It is just a point to make here that, unlike other products that go through a standard pre-qualification pathway, it was a slightly different issue with the WHO with the RTSS approval, and that RTSS is actually now listed among the pre-qualified vaccines. So I just wanted to make that point for clarification purposes, and we are happy to convey any additional questions to WHO. Adrian, I hope that is perfectly okay with you.
Thanks, Pedros. I didn't, Pedros, I didn't know that, because it happened 2 weeks ago. I can see the date on the WHO link. So Cynthia, thank you for sharing that. A little bit surprising that there was massive New York Times publicity of the policy recommendation, and certainly to my knowledge, not very much press about the actual pre-qualification. So maybe that's to come. Okay, thanks for sharing that.
No, I think that's great, and it's, we are grateful to our colleagues listening. This is also one of the reasons we do these masterclasses openly like this. I think it helps with that. So thank you so much for that. Let us move forward. We are coming towards the end, so I would like us to move really towards the very end. Adrian, we've talked a lot about vaccines. At some point, we might ask you to talk about capacity building, but let's keep that for now. A very brief commentary that came from your group, but which we found very important, was this brief piece in the Lancet where you described the reasons that you guys are happier. And this is by Datu et al. As well. Why you think that the future is brighter, and I think we've alluded to this. And here you talked about, first of all, of course, the studies from Burkina Faso that combined SMC plus RTSS. You talked about the R21 studies, and you talked about monoclonal antibodies. Now, a brief commentary from the Gates Foundation by Philip Welkoff, who is taking this head on and is very excited about potentially transformative tools, has emphasized the fact that we need to expand and enhance the innovation platforms for malaria, just to make sure that we have new tools that can be used together. And here is from the Lancet Commission on Malaria, a number of new tools that were highlighted there. One of which you guys are very excited about as well, monoclonal antibodies. Here was the first publication that came from this group of CEDAR's group last year, monoclonal antibodies demonstrating something that is pretty powerful. And just this morning, they published a new paper on low-dose subcutaneous and intravenous monoclonal antibodies. This is out in the New England Medical Journal today, I believe on August 4th. This morning, it's got a fantastic commentary from MMV by Dr. Tim Wells that explains all these and why this particular candidate is fantastic. Now, of course, monoclonal antibodies are not vaccines, but they do have some indications or use cases that are very similar to either SMC or vaccines. And I would like to hear from you broadly, and Ali, it would be nice to hear from you coming to this as well. What are your thoughts about monoclonal antibodies for malaria vaccines, for malaria control? How would they play into the landscape that is that already has RTSS, or as instead of vaccines or in addition to vaccines? Generally speaking, how is this going to transform malaria control? Sorry for speaking a little bit too long on this one, but this is another very exciting intervention technology that is coming up. Back to you, Adrian.
Yeah, I mean, it's the same problem as with scenario sporozoites. You can't get the cost of goods down into dollars rather than tens of dollars a dose today. And, you know, there's still hope that you might be able to improve the manufacturing process. But remember that billions of dollars have gone into improving manufacturing yield for monoclonal antibodies. And if you want to have a monoclonal antibody to help cure your cancer, the cost of goods is not tens of dollars. The retail price is thousands of dollars. So I think, you know, for very rich armies, this is quite exciting. So if you're the Pentagon and you want to make sure that you have something that works to fairly high efficacy, it doesn't look 100% in that paper, by the way. But, you know, if you want to give your soldiers a shot before they go into a malarious area to war, this is certainly quite exciting. In terms of cost of goods for routine childhood administration, if monoclonal antibodies worked, we'd have RSV antibodies being given to children in low-income settings, because RSV is an important pathogen, and we'd have it being given to older adults, and that's not happening at the moment. So if we can't give monoclonal antibodies for RSV to elderly Americans who can definitely afford quite expensive healthcare interventions, I'm not quite sure we're anywhere close to giving these routinely to every child repeatedly in low-income settings, even though the science is great, and Bob Cedar has done a lot of work to get it to where it is.
That's interesting. I had a very different opinion on this. You know, I thought it's going to be great, and I thought that, you know, we can, you know, it's just a matter of technology being early, and that with scale that you could actually cut the prices.
With scale, you will cut any price, but you've got to have somebody who's spending billions of dollars already to get it up to scale. So yeah, you know, there's a reason that monoclonals cost more than vaccines. Right. They're harder to make. Right. And by the way, they are single epitope targeted, but in this case with NAMP, because you have lots of NAMPs in the parasite protein, this is a good starting point to try and get monoclonals to work against malaria, because it's unlikely the parasite can escape from that immune response. But that's the same reason you're using them for vaccines.
Excellent. Ali, do you have any different thoughts?
No, I think it's fair to learn from Adrian's comment. I think we have to be careful and recognize some of the challenges or the hurdles that face the technology or the process, you know, before we can have the vaccine in the field. So I appreciate that what Adrian has said. But really, as a concept, it really looks great. It sort of addresses some of the challenges perhaps we have with the vaccine in terms of being able to, you know, inject a significant amount of antibodies, and if these are sort of long lasting, they could have an impact from preventing, you know, from in reducing the risk of malaria infection. And they will definitely, if they are to be deployed in the field, despite the challenges that has been highlighted, I think it will add a significant, it will have a significant impact in terms of preventing, infection in children. It can be used, as you rightly said, maybe as SMC in a SMC setting, but it could also sort of be used in early childhood just to sort of protect vulnerable children, pregnant women, etc. So it could be very helpful in high risk population and in different settings, especially in areas of very high transmission, high commodity transmission. So as a technology is great, but I think all those challenges that have been highlighted by Adrian are something which we should think about. But more importantly, I think we need to ask these questions. I mean, when we are now embarking in evaluating this kind of product, we need to think about all these challenges, and not only deployability, but also this be available sort of for widespread use. So I think this is an important point that Adrian is addressed, is highlighted here.
It's a part of the deployability question. Thank you both very, very much for this. Adrian, we have only 2 last questions for you, and I'm going to post both of them, and you can answer them in whichever order. Then we will flip through the slides as we come towards the end. Number 1 is the question of combinations of vaccines. One topic that you yourself, as many other people have proposed a lot, is the idea that you can combine, that if you combine vaccines, if you put vaccines of different stages, if you targeted multiple stages of the Plasmodium falciparum life cycle, that you could get greater efficacy. Based on some of your studies, it suggests that it's not a simple question. It's an important question, but it's also a question of how the protocol is, how exactly you deploy it. It might be that we are misunderstanding your work here, so it would be nice that you clarify that. So that is point number 1. In one of your early reviews from 2011, actually, you emphasized the need to consider this and evaluate those ones as well. And you actually did state here that many reviews of malaria vaccine have proposed that the ultimate malaria vaccine will be a multi-stage and multi-component vaccine, providing results of protection in genetically diverse human population. But then you also highlight some potential challenges to that. So that's point number 1 that I would like us, the first of the last 2 questions. The second is a topic that I first heard about when I read work on the malaria research agenda, where it was proposed that actually fractional doses of vaccines like RTSS might give you better value than the current ones. That the idea of low doses is something that has been neglected, but it's potentially very useful. And you guys have done some tests on that. You have one fantastic study that is recently out of your lab, published out of your lab here, that indicates that a delayed fractional dosing with RTSS actually gives you some great results as well. So in whichever order you would like to take these 2 last questions, I'll be flipping through some of the data just for our audience to look at. It would be nice to hear what your opinions are on these 2 questions. Number 1, multi-stage processes, and number 2, fractional doses for this vaccine. Back to you, Adam.
Great, thanks. So let's take the bad news first. Delayed fractional dosing does work in adults. Been shown repeatedly with RTSS, with RH5, again by Simon Draper's group. If you delay the 3rd dose, 0.16 rather than 0.12 months, and reduce it by, say, 5-fold, you can get not only higher antibody titers, but more durable antibody titers with some antigens. That's the good news. The bad news was that's only been tried once, to my knowledge, in African children with RTSS. It was reported late last year at TropMed that essentially the efficacy of RTSS, whether you give it at 0, 1, and 2 months in children or 0, 1, and 6 months with a fractional third dose, is the same. So why it works in adults and not in the key target population for malaria vaccine is unclear. But obviously people should carry on working on that. My guess as to that answer, for any of you who want to try it, is that RTSS has been overdosed for years. I mean, 20 years. Why do you need 25 micrograms of RTSS and you only need 5 micrograms of R21? There weren't, to my knowledge, significant dose escalation and de-escalation, particularly trials in West Africa way back in 1997, 98, 99, when it first went to Africa. Maybe it's time for somebody else to do those in Africa and see if 3 fractional doses of RTSS is the same as a 3rd fractional dose. Why is that important? Well, you now know it's because there's so little RTSS around. If you could get 3 doses out of 1 dose, it would be fantastic. Somebody should try that. Please, please do. And it has to be in African children. Okay, then to finish with, as you say, multi-stage vaccines. Yes, absolutely, definitely. It's the final answer. We need to do those. That's just one of the reasons to remind you why an anti-NAMP antibody won't be as good as vaccination, because in a few years' time, we will be putting together R21 vaccines. And remember, GSK won't do that because they're out of vaccines. They're selling off their vaccine to a biotech in India. They're not making blood stage vaccines. They're not making transmission blocking vaccines. In Oxford, we'll put our RH5 vaccine together with R21 this year in the Gambia, next year in other countries. Yeah. In addition, we will add a transmission blocking component. We haven't talked enough about transmission blocking vaccines that are going to be extremely important in the end game for malaria elimination and eradication. There are 3 antigens that are in play. We don't have 3 antigens for sporozoites, there's only 1. We don't have 2 antigens for blood stage, there's only RH5 that has shown efficacy. We have 3 antigens for transmission blocking stages that show high functional activity in the key functional antibody assay. And we even have a first trial, only 1 trial, looking at real efficacy of transmission of parasites to mosquitoes from humans. That was 74% efficacy with PFS 230. That doesn't mean it's the best antigen. It does mean it looks as if transmission blocking antigens are going to be relatively easy. So I'm super optimistic about combinations. You can be unlucky. Antigens can interfere with each other. I think that's unlikely, but is going to be tried soon. So, you know, I think that's a very positive message to finish with. We're going to be able to make multi-stage vaccines that will be almost as cheap as R21 with potent adjuvants. We now know what the best adjuvants are. We know they're safe in African children, at least so far, and we'll be able to move forward rapidly. So, you know, we've got to stop thinking about malaria vaccines coming in 5 years. It's nearer 5 months for several vaccines. And we need to press our regulators and public health authorities to be a bit more demanding of international organizations who say they're supporting malaria. The time to support it is now, not in 3 years' time or 10 years' time.
Right. Ali, you work on RH5, and you will be working on R21, and you have worked on RTSS. And I was telling Adrian earlier that at Ifakara we have he said I should correct it, but you know, we thought that history has conspired to have us test every malaria vaccine candidate ever tested, almost, starting with PFSP66. I hope our director always corrects me when I mention this, because I always misstate the name of the Colombian vaccine. What is it? SPF66. So we tested that. We tested the RTSS with the AS01, AS02. We tested. We've been testing Malaria vaccines. We're testing RH5. Ali is working with you on many of these things. So, you know, Ali, what are your thoughts about these combination vaccines? And I know I've asked you before, but when I first asked you guys this, I wasn't very happy because I thought you were going to tell me, you know what, we need a vaccine and then a good vector control tool and then a good case management tool. But then you guys said, no, we need a vaccine against blood stage, and then we need a vaccine against liver stage, and then we need another vaccine against I don't know which stage. And I'm thinking these vaccine people are nuts. You know, they should be talking about broader integration of these tools. What are your thoughts about these multi-stage vaccines as an integrated malaria control tool vis-a-vis multiple interventions rather than just 3 vaccines put together? Back to you.
Pedro, yes, thank you for that. No, I think you're putting me on the spot. I think it's, I mean, again, I've worked on vaccines for most of my career, so I definitely also agree with Adrian. I think a multi-stage vaccine would be a great thing to do because, again, the aim is to improve on the efficacy, and we've talked about each antigen is sort of adding on and improving as you know, new technology comes in, and it's the same. So I mean, combination is part of trying to come up with a vaccine that can increase sort of efficacy in terms of, you know, protecting individuals against you know, disease and transmission. And it's kind of a way to ensure there are multiple obstacles for any breakthrough parasite, which, you know, bypasses one of the immune responses induced, etc. However, having said that, I will go back to our earlier comment that Adrian made, and that is, I don't think we're talking about replacing other interventions. I think it's always about you know, doing this on top of other interventions, and I don't think you know, thinking about now as we're developing these vaccines, thinking about, you know, the vaccine will come and replace all the intervention. That is the right way of thinking. So I do support that idea of coming with the multi-stage vaccine. It's a great thing, I think, in malaria control, but I think should still continue to strengthen the other control methods that are available in the field. Thanks.
Thank you. Thank you so much. I don't know how to say this, but you know, we have always, as always, many more questions. We are having to cut this short because of time. We had many questions for you, Adrian, on severe malaria. For example, I see your colleague Alice is on the call, with whom you've done some fantastic studies on, for example, relationship between severe malaria and iron deficiencies in Kenya. I don't know if I'm understanding, maybe not Alice, but one of your colleagues from Kenya. You've done some great studies on that. And Alice and Ali and the rest have also done some great studies on severe malaria on that side. And we had some questions on that. We're having to bank those ones for a future masterclass. We had some questions on capacity building, but I think that looking at the profile that you have built over time, the relationships that you've built, the infrastructure that you're making available, it's a testament to just the capabilities that you're helping build around the world. And again, like I said in the beginning, on behalf of my colleagues and everyone on this call, we are very grateful to you and to your partners. Before we end, though, I would like to ask you, Adrian, what your thoughts are, if you have any specific questions for us, if there's anything that we didn't cover that you would love to at least mention before. So in other words, you have the last word. If Shayla is willing to take the last word, she would, but she is on the call as well. And then Ali, and then we can close. So back to you, Adrian.
Yeah, so firstly, my thanks for your inviting me. It's fantastic to have the opportunity to speak to this super interested and very experienced audience in malaria and many other diseases. Really, all I wanted to say was, look, I'm delighted by the momentum that COVID has unwittingly generated in Africa on all things to do with vaccines in Africa. I don't just mean accelerating vaccine supply for malaria. I mean manufacturing vaccines in Africa to become self-sufficient in that area, to increase the pipeline of African vaccines in Africa. I think that's been a long-held aspiration of people like me who are trying to build capacity through our vaccinology in Africa courses and so on. And it's fantastic to see more and more young African scientists demanding that they have access to this capacity. It's ridiculous having a lot of it done thousands of miles away. So my congratulations to everyone who's involved in this renaissance, if you like, of vaccinology in Africa. Please keep the momentum going. Money is coming in faster. It's a long way to go, but it's the right time, and the very best of luck in pushing this forward. Thank you.
Thank you so much, and we hope that you enjoyed the masterclass as much as we did, Adrian. Definitely.
Great audience, great questions. Thank you.
Thank you so much. Ali, do you have any last words for Adrian?
Not really. I think I would also like to thank Adrian for agreeing to participate in this masterclass, and we definitely take his recommendation to push, keep on pushing the effort. I mean, pushing the this great wheel forward. Yeah.
Shayla, please, are you still there?
Hi, Fred. Yes, I'm still here. Hello, everyone. Hi, Adrian and Ali. My apologies as well. I wasn't able to join in time, but I'm happy that I joined towards the tail end, and I got to hear about where vaccine comes from. It actually comes from trees, that I know, and to also hear from you, Adrian, about the many opportunities there are for especially our students who attend these master classes on the topic of vaccinology, vaccines and malaria. So I was really glad to hear about this. Just thank you to all the participants for attending today, and for our host, to Professor Adrian. Yeah, thank you, Fred.
Thank you, guys. Thank you so much. We're going to bring this to an end. Very, very grateful for your time. Sorry about the level of presence 2.0 is demanding his time now. But do have a wonderful time and enjoy the rest of the day. Our next masterclass is going to be on chemo prevention, and this is going to be in line with the recent WHO recommendations on chemo prevention. So we are aligning speakers for that. Our colleagues in Tanzania have been doing some great trials that they would also like to share with us. And the first time we're going to have an NMCP specific masterclass focusing on one specific country, and this is going to be piloted with a masterclass from the NMCP Tanzania. So once this is scheduled, we will send you guys an announcement. But lastly, just want to say thank you so much once again for participating in the event today. Thank you, Adrian, for bearing with us. All the 3 hours, we started with nearly 200 participants. At the 3-hour mark, almost 3 hours, we still have nearly half of the people on the line still, and it's also being watched live on YouTube. We are grateful for that. Thank you so much. May you have a wonderful day. We're going to bring it to an end right now.
Thank you.